Проверка раз-два раз.
Раз, раз, два, три.
Зажимайте, зелененькая горит. Да, да, да. Спасибо.
Раз, два, раз, проверка связи, раз, два.
Подожди.
Может включить в зуме, раз, два, один, раз. Проверка связи.
Может, у тебя там что-то?
Один, раз. Все, я звук в зуме выключаю. Так я уже разрешил. Тебе надо права разрешить запись локальных файлов, да? Ну сейчас пробую.
Эта конференция записывается.
Все, окей.
Давай, что?
Good morning, everyone. I would like to start our second day of the conference. From what I've heard, the first day went really well. Oh, okay. Thank you. So, good morning, everyone. I would like to start the second day of our conference. As I've heard about the first day, mostly the opinion is positive, and the participants, some of them I managed to communicate with, expressed their positive feelings about the first day of the conference. Today we have two sessions, and I'm presiding one of them, the animal probiotics, and I would like to give the floor to our first speaker, Rivaz Lapachi from the Agricultural University, the campus named after Kaha Bendukidze. Rivaz, good morning. I can see you are with us, and the floor is yours. We're waiting for your presentation.
Good morning. Can you hear me? I will share my presentation.
Yes, we can hear you. Thank you.
Yes, he.
Окей. Доброе утро, в первую очередь, всем. Приятно, что я сегодня с вами здесь. Наше приветствие с Аграрного университета Грузии. I will follow on English. Thank you very much for possibility to be a part of the conference which you organized. I will talk today about our small experience.
I'm sorry, can you please talk a little bit louder?
Louder, okay,
yes,
I
you.
can. Today I will talk about Bacillus subtilis and its impact on broiler. We make some small researches on local Georgian conditions. I will make some presentation of our experience on local market and challenges which we faced in Georgia. And then we can also discuss the problems in Georgia and our methodologies, how we work and how we see the beneficial microbes in our life and also in life of farmed animals. First of all, I will start with an introduction of the project which we are running now, and thanks to Shota Rustaveli National Science Foundation of Georgia, we are running this project, and with the supervision of Ambrosic Kwaseli, the professor of Animals Husbandry of Georgia, and Michael Stiani, PhD of Institute of Microbial Biotechnology. Our constant is Vladimir Elisashvili. He's director of Institute of Microbial Biotechnology. Our team is represented with the next specialists, and we have quite broad specialties in our team, and we are supporting and helping each other to make our research and studies more effective. In general, and in few words, the problem which we are faced now in Georgia, as in big integrations and in small integrations, we have uncontrolled, there is a lack of knowledge how to use antibiotics, why to use antibiotics, and what could be the alternatives of the antibiotics in the poultry industry on Georgia. I will talk about the strain Catmira, the Bacillus subtilis, with the exceptional probiotic potential which we test on broiler. We will talk about the inclusion rates and about experimental details which we set on local integrator roster on Georgia. And generally, I will talk about the challenges of the world, and we, Georgia, as the small part of the world, faced with similar problems, more or less, sometimes much more problems with more developed countries with antibiotics residues in meat, with problems with resistance to the antibiotics by the different pathogens. On Georgia, we broadly use different antibiotics in not only for treatment, but also for prevention of different diseases. And we have some growing demand of the quality and what should be the meat quality for the consumers. Several alternatives, of course, are available in Georgia. Different companies are working very actively to introduce some of them, and I will talk about what kind of alternatives are available now at this moment. If you will see from the broad figures, this is the data from 2017. This is more closer data which was available for us for today. If you will see the more developed countries, the average consumption of meat, which is containing beef, poultry, pork, and lamb, is 80 kilograms per person per year. And in Georgia, we have still very low consumption of feed, even on Azerbaijan and Armenia. So we have still the room to grow the sector of the poultry, because the challenge of how to support our local poultry production is the open agenda even for the government. So we are trying to support from our side the poultry integrators with the solutions of gut health and supporting them with alternatives of some treatments, such like for antibiotics treatment. In Georgia, you will see the balance sheet of the meat consumption is something like this. You can see the lamb consumption on the blue, beef consumption, pork consumption, and poultry consumption by the years. So you will see that poultry is most common meat on Georgia because it's cheap and more reliable on Georgian market. And it's going up from year to year. And we will work not only for the poultry, but we are now working also on the rabbits direction. And of course, we will think about how to apply some special probiotics to the pork and beef industries also. About the mentioned alternatives of probiotics which we are faced in Georgia, and me as the nutritionist who is working with the farm animals nutrition, we have several alternatives for probiotics, organic acids, minerals such like zinc oxide, and even we now start trials on the spirulina, some fags and plant extracts. But anyway, these are the local problem solvents. So these are not general problem solvents such like antibiotics, which are broad spectrum activity and could work on different systems such like urinary systems, such like respiratory system or gut system, gastrointestinal system. On these concrete tasks, we are working on the gut health and intestinal health, and to creation some natural barriers against the pathogens. So why we choosing Katmira for this moment? We had some special activities and pre-research made of agricultural university team, which was conducted and investigating the regulations and improve production of probiotic spores on locally. Why? We were thinking about the alternatives, about the prices, and about the possibilities to product some cost-efficient and effective products locally. Because we have, from one side, we have scientific basis how to produce the probiotics, and on the other side we have cheap wastes such like agro-industrial wastes, wheat bran or molasses or vinasse or GDGS, which are very good substitutes for bacterial spores cultivation. So that's why the first research made on 2015-2017 give us possibility to make new experiments and trials on effectiveness of Katmira on the broiler productivity, which was set on 1 year under this project which I mentioned above. If you will see roughly the raw materials listed in the example ration of the broiler, we can use up to 10% of the raw materials which are used also for the feeding of the subtilis. Wheat bran, corn DDGA, some molasses, vinasse also. So this is very good product for production of the probiotics, but it also works as the similar nutritional nutrient for the probiotic in the intestines. So our goal is to have strong strains which will be able to work on the technological lines of the feed mills, such like they must be strong enough to face with thermal impact from the granulation process, and also they must be livable in the gastrointestinal tract. So they also have some nutrients in the feed, and also in the intestine they are available to be more effectively grown by the nutrients which we support them. The Katrina was tested on the solid state fermentation. We defined the concrete concentration of this product, and we use 3 different inclusion rates of the final feed. We use 0.03%, 0.04%, and 0.05% in the total feed as the feed additive. And we introduced this feed with this additive to the broiler chicks. The trial schedule looks like this. We have 4 groups with 100 on each, totally 400 poultry, 400 per birds in the trial. And I will also mention that we are working parallel with these trials with the rabbits also, and we are working on second strain, which is Bacillus amyloliquefaciens, and our trials is also conducted with the Bacillus subtilis and Bacillus amyloliquefaciens in the combination with each other. But today I will not talk about these directions. I will talk about the Bacillus subtilis and its inclusion in broiler and its impact on the broiler productivity. We set the trials on broiler with help of our friend integration company, which produce broiler and support local market with the broiler meat. And the data which we gather from the field give us possibility to understand and know how the Bacillus subtilis affects the productivity and performance objectives of the poultry. So we said that 0.04 grams per ton of complete feed is more or less most effective from our point of view. And we test some feed conversion ratio, average daily weight gain, absolute weight gain, and survival rate with productivity index, of course. With our trials, we also make some experiments and trials on the blood, meat, and intestines, of course, to check some deviations or some problems with the analysis which we made. So if you see, we have 4 groups. One is control group, this one is test group, second one, third one, and fourth one. We have different inclusion rates of the probiotic in the complete feed. And generally what we see from the 1 condition poultry, 1 age poultry, which was 1 parent stock chicks with more or less similar body weight gain, we see that we have more livability in the test groups, all test groups. No, anyway, 5% of mortality is not a problem in Georgia. We have even more mortality in our poultry industry because we are suffering from such problems such like salmonellosis and mycoplasmosis on the poultry. And we also have some problems related to viral diseases. And generally the problem on Georgian poultry market is that we have some multi-age farms, and the problems related to the biosecurity is very high. But on these trials which we covered during this experiment, we see that on all experimental groups, survival rate was much more higher than in control group, and also the body weight gain and absolute gain was a little bit higher. This was not affected by the FCR, and feed conversion rate was not significantly changed. But anyway, by our trials, we can say that in the conditions where poultry is suffering with restricted movement and artificial environment, so any feed additive which have beneficial impact on the poultry could work very good. And there is some open questions which must be covered related to the shelf life of the probiotics, how much can we store it, how can we use it during the heat treating on the production lines, I mean feed mills which producing the feed, and how to use it and what are the appropriate doses. And these are the tasks which we are running now and we are trying to explore. But anyway, from the figures, we can see that even on first, 14th, 80, 28, and 35 days, we have higher life weights on all groups compared to the control group. We have absolute daily weight gain also increased by 7.3%, and this is not small amount, not not so much, but also not too much, too low, because in big integrators any plus is a big plus, and daily gain with livabilities was very exciting for us. So if we will look from the comparison of the price and cost, we are now on the stage when we are calculating what could be the cost of the commercial product produced on locally, I mean Bacillus subtilis, and we will compare it to the antibiotics treatment, and also we'll be able to compare two products, because antibiotics are not fully replaced in Georgia, and we will still compare the price segments of the premium or more premium meat, because there is some brands which are not treated at all with antibiotics. Someone have some demand to don't use GMO raw materials. Someone want to say that this product is antibiotic free, and this is also opportunity for us to make new value on the market, to introduce products which will replace the antibiotics but will also have beneficial impact on the productivity of the poultry. FCR, as I already mentioned, have no big differences from each other, and productivity index in all trial experimental groups compared to the control groups was 24, 34 points higher than in control ones. So what we can say shortly about our trials, which are going around a year and a half, we can say that probiotics on the local agro-industrial waste, which are produced locally, could beneficially work on fields and on broiler integrations and farms. And we can say that we can replace antibiotics on several conditions, not in all, and have better results on survivability and productivity. This is the field work of our team, where we conducted the data from the field and from the integrations. We were making some control of the environment, control of body weight gain, development, feed consumption, water consumption, management system of the farm, everything. We make the sample preparation of the intestines, blood, and meat, of course, to check different possible deviations which could rise during the experiment. We make some trials and experiments on the laboratory of microbial of the Institute of Biotechnology with Vladimir Lisashvili. So these trials are connecting two institutes of agricultural university, and now we are on the way to make some commercial offers to the big integrators of Georgia and make this product available to the local market, because the trials which we conduct and which we made on two species on poultry and rabbits with different inclusions and the combinations of probiotics give us hope that on some conditions, probiotics could work very beneficial on the market. So this was shortly what I can share to you for today. And we will continue working on this project, and we will share all the data if this will be interesting for you, of course, with more details and more results from our experiments.
Thank you very much for your presentation. It was a particular pleasure to see it today, to hear it today. And I think many of us know that our two groups are collaborating, and this is really many years of fruitful, most enjoyable collaboration, which resulted in several good quality papers published, and I'm very happy to see that right now this project is alive and developing. We have time for a couple of questions. Yes, please.
Hello, you said about very large number Bacillus coloniforms units per gram, 10 in 12th degree.
Yes,
what
yes.
methods you use for this Bacillus quantity?
You mean the method of the cultivation, yes?
Yes.
Yes, as we work on the laboratory, we use a solid state fermentation method. We try to receive as concentrated colonies as possible, and also with the high index of 10, 12, you mean, yes? We are trying to make with this also the effectiveness, but also the cost effectiveness, because as concentrated is the product, as a low inclusion is required to achieve in the final product, I mean for the producers or the feed millers. So the most important what I know from the supervisors and the consultant from the Institute of Microbial Biotechnology, that they very hardly work for 5 years to reach these figures, to have as much concentrated spores in 1 gram. So this is one of the benefits of our probiotic compared to the different commercial products available in Georgia also. So that's why I mentioned that this is a unique product which is cultivated by the support of 2 universities, also from Rostov and of Don and Georgia Agricultural University. And our team of microbiologists are hardly work on that direction.
Thank you.
Thank you for your question.
Any more questions from the audience?
Like one I have,
Yes, please.
if you allow me.
Dr. Santosh.
Otherwise, audience will be first, I will be later.
Go ahead.
Nice to see you all again, and thanks, Reji, for presenting a very nice work. Actually, really, this is a demand of time. I have some of the questions, but would be very fast. I would like to interact with you that what is the growth response in the solid state as compared to the liquid state of this bacillus? Have you compared if you grow in the liquid condition and like you have used the solid state, that's fine. But we have seen some of the literature where the growth profile is entirely different than the liquid state.
Yes, thank you for your question. At this moment, our trials for Bacillus subtilis as Bacillus amyloliquefaciens are set only on solid state fermentations. We don't compare it with liquid form fermentations, but we have it on mind, and we will
Yes, my concern was only to optimize the conditions so that you can generate large amount of biomass and it will be helpful.
Hello, can you hear me?
Yes, yes, you are audible.
Sorry, I have some problems with connection. Yes, we have on mind to compare these two methods of fermentations, and we will make the further trials on that also. And I will have consultations with Mr. Vladimir Listashvili on this direction also. Thank you for your remark. This will be also interesting for comparison. Thank you.
My suggestion would be to optimize the conditions under various parameters. So you can use different, we have statistical tools for statistical optimization. So you can have a lot of parameters and optimize best condition. Though another would be, have you checked this with other probiotics like lactobacillus or enterococcus, such kind of growth in solid state or only on the bacillus?
What I can do for this, if we will exchange the contact data, we can use your suggestions, and our two institute with collaboration of with you also, and to receive your recommendations, we will set different trials also, because on Georgia now we are free and open to all new possibilities to cultivate microbial supports and probiotics. So any novelty on any idea from your side or any side will be more than welcome. And
dear colleagues, I want to ask you to keep it short. We still have to stay on time. Yeah. I really appreciate this very, exciting discussion, very encouraging discussion. And just to finalize this first presentation, Dr. Santosh Tiwari is also collaborating with our group. And if I'm not mistaken, you used wheat bran as a substrate. And we have experience of working with these strains using different substrates, which are either byproducts of agriculture or byproducts of production of food products. I think this is a very interesting direction, and what Dr. Tiwari mentioned, moving to lactobacilli can be a very interesting ground for starting international collaboration. Thank you very much. We still have to keep on time, and we're a little bit behind now, so I will appreciate everybody being on time with their presentations and not to go over. The next presentation today is from Dr. Jane Sang Lin, Director of the Culture Collection and Research Institute of Sunbird Tech from Taiwan. Dr. Lin, the floor is yours.
First of all, I have to thank the organizing committee to give me the chance to share our research study. The topic of today I want to share is Lactobacillus plantarum TWK10 attenuate age-related muscle weakness, bone loss, and the cognitive function decline via modulation of gut microbiota in mice. According to United Nations report in 2017, the number of older persons is growing faster than the number of people in all younger age groups. In 1980, children aged 0 to 9 years substantially outnumbered persons aged 60 years or over. But by 2030, the global population of older persons is expected to have surpassed that of children under age 10. The projection also indicates that in 2050, there will be more older persons aged 60 or over than adolescents and youth at age 10 to 24 years. The number of people at very advanced age is increasing too. The global population aged 80 years all over is projected to triple between 2017 and 2050, increasing from 177 million to 425 million. However, an increase in advanced life expectancy does not necessarily mean an increase in healthy life expectancy, but also include the persistence of various disease and complications. Many functions of our body decline as we age, especially on brain function, cardiovascular function, lung function, muscle function, body composition, and metabolism. A healthy intestine is under homeostatic condition. The epithelial cells produce antimicrobial peptides in response to interleukins and also express pattern recognition receptors. The gut microbes regulate mucus secretion and the AMPs production and also regulate the gut barrier integrity by producing short-chain fatty acid. Gallbladder cells produce mucus to constrain pesticides invasion. Lymphoid cells play a role in host defense by producing controlled array of interleukin-22. Dendritic cells induce the activation of differentiation of naive B cells to produce plasma cells that produce commensal-specific IgA in the lamina propria. IgA is transported into the gut lumen and binds to commensal microbes and soluble antigens, thereby restraining their adherence to the host epithelium and the leakage through the gut barrier. However, under dysbiosis or senescent milieu, the altered microbiota composition and the weakened gut permeability may lead to increased adherence and the leakage of various microbes and the microbial byproduct through the gut barrier, thereby instigating hyperinflammatory responses, eventually increasing the host susceptibility to various gut-related health problems. In recent years, many researchers have been published to demonstrate the potential use of probiotics on anti-aging, like skin aging, immunosenescence, muscle loss, cognitive decline, and longevity, etc. Or from our previous published animal and human studies, we found that supplementation of Lactobacillus plantarum strain TWK10 can improve exercise performance, muscle strength, and also muscle mass. This result inspiring us to further study if these effects will still work on aging individuals to further prevent aging-related function decline. Then we conduct a study to evaluate the impact of TWK10 on muscle loss, bone health, and cognitive functions in aged mice. We also evaluate the impact of this probiotic on gut microbiome to study the potential relationship of microbiome and the aging process. The muscle mass and the muscle strength increase as we grow up in our early life, but the trend reverses after 40 years old. We began to lose our muscle mass and the muscle strength, and those up to 8% per decade. As we get older and older, the loss rate accelerates and almost doubles to up to 15% per decade. The aging-associated loss of muscle mass and muscle strength, we call it sarcopenia, is closely related with increased risk of falls and fractures, physical disability, poor quality of life, and even death. To study the impact of lactobacillus plantar TWK10 on age-related male dysfunction, young and naturally aged mice were used for comparison. Both young and aged mice were divided into two groups and administered with probiotics or vehicles, respectively. The dose of probiotics was 10 to 9 CFU per day per kg. The duration of the experiment was 8 weeks. We can see at the beginning, the muscle strength of Asian mice was presented by grip strength was significantly lower than young mice. After 4 and 8 weeks of the experiment, the grip strength in young vehicle group had no significant difference. However, in aged vehicle group, the grip strength declined significantly compared with zero and week zero. Administration of TWK10 improved grip strength both in young and aged mice. We can also see the gastrocnemius muscle weight significantly decreased when mice aged. But we didn't see any significant impacts of TWK10 administration both in young and aged mice. But the glycogen content in the muscle was increased by TWK10 administration. And we also found a positive correlation between muscle glycogen content and muscle strength, no matter in young or aged mice. Similar with muscle mass, bone mass declines as we age. Peak bone mass was achieved at around 30 years old, and then begin to lose 0.51% per year. Bone loss is more severe in women after menopause at a loss rate of 1 to 2% per year. Bone loss is called osteoporosis, is the condition that causes bones to become thin and porous, decreasing bone strength and leading to increased risk of fracture. To evaluate the efficacy of TWK10 in restoring bone quality, marine female bones were harvested after 8 weeks of treatment, and the cortical and trabecular microstructures were analyzed using micro-CT. Age-related bone loss was demonstrated as a significant reduction in trabecular number and an increase in trabecular spacing when comparing the Y control, the young control, and the age control groups. The bone fraction expressed as BV/TV values was significantly higher in the young TWK10 group compared to the young control mice, whereas the value of TBSP in the young TWK10 group was significantly lower. Administration of TWK10 resulted in a significant increase in the trabecular number of young TWK10 and aged TWK10 mice compared to the control mice. And very interesting is the serum level of vitamin D in the aged TWK10 was also increased compared with those in the aged control group. To evaluate the effect of TWK10 on aging-related decline in spatial learning and memory, the Morris water mass test was carried out. The mean escape latency of the aged mice was significantly higher than that of the young mice. The mean escape latency of the young TWK10 group on day 2 and day 3 were significantly lower compared to that of the young TWK10 group on day 1, whereas no significant differences were observed at any of the time points in the young control mice. The differences in the mean escape latency between day 1 and day 3 in the aged TWK10 group was significantly lower than those in the aged control group, whereas those in the young TWK10 group showed a decreasing trend compared to the young control mice. Short-chain fatty acids such as acetate, propionate, and butyrate are important metabolites in the maintenance of intestinal homeostasis. We therefore investigate whether TWK10 could facilitate the production of short-chain fatty acids through the modulation of gut microbiota in young and aged mice. The concentration of acetate, propionate, as well as butyrate in the cecal content of young TWK10 mice was significantly higher compared to those in the young control group. In young mice, we found that the administration of TWK10 results in higher abundance of short-chain fatty acid producing bacteria. In the case of acetate producing bacteria, the abundance of genera Prevotella 9, Luminoclostridium 6, and Luminoclostridium 9 in young TWK10 mice compared to those in the young control group. The abundances of butyrate-producing bacteria, the family Pyracea, and the genus Spiraceae, UCG-006 was significantly increased as a result of TWK10 administration, and the significantly increased trend were observed for members of the family Luminococcia and the genus Roseburia in young TWK10 mice compared to those in the young control group. On aged group, the second short-chain fatty acid in aged group no matter in short-chain fatty concentration or the microbiota has no significant difference. Finally, I have to conclude from the above result, TWK10 ameliorate age-associated muscle weakness and cognitive function deficits in aged mice. Further, age-associated bone loss was also potentially attenuated by TWK10. TWK10 modulated gut microbiota toward a better profile by enhancing the abundance of short-chain fatty acid producing bacteria. TWK10 could be the potential therapeutic agent for attenuating age-related disorders and promoting healthy aging. Finally, I have to thank Dr. Huang and their team in National Taiwan Sports University for conducting most of the experiments, and also our team in Symbiotech. Special thanks to Dr. Watanabe for his help in microbiota analysis. Thank you.
Dr. Lin, thank you for presentation. Can we check if Dr. Lin is with us today to ask questions if he is with us? No, probably he was unable to join us. So if anybody has any questions, please forward them to the conference organizing committee, and the questions will be passed to the presenter. And we're moving now then to the next presentation. So he's not here. He's not with us. Его нет с нами. Right, and we're moving to the next presentation. Dr. Mazenko, the floor is yours.
Hello, dear colleagues. On the PubMed website, at first glance, it may seem that the problem of finding new probiotics does not exist. Indeed, the number of articles dedicated to these topics increases every year. However, at the moment, is it scattered information where there are no general approaches to finding probiotics, testing them in vitro and vivo, selecting strains and comparing them with each other. This makes it difficult to select strains to create new drugs for medicine and agriculture. We propose our approach to finding new probiotics bacteria and creating drugs based on them. First, searching for probiotics bacterial strains in organisms of the same genus and species for which the probiotic product will be created. This slide shows sampling points for hens with different types of housing. As a result, it was isolated 19 strains of Bacillus and 38 strains of lactic acid bacteria. Second, safety check. The isolated bacteria were tested for mutagenicity, hemolytic activity, antibiotic resistance profile. None of strains showed mutagenic or hemolytic activity. 4 strains show resistance to antibiotics and were excluded from future studies. Third, in vitro study of potential probiotic properties. We studied antioxidant and antimutagenic properties to evaluate the potential probiotics properties of the strains. We used Lux biosensors, which are recombinant E. coli strains, contain plasmids with photobacterial species with luciferase operon, but under control of corresponding E. coli promoters. Biosensors with the picket G plasmid respond to the presence of oxidants, in particular hydrogen peroxide in the environment. Biosensors with pyrrhic A plasmid respond to the presence of DNA damaging factors in the cell. This slide shows antioxidant and antimutagenic properties of Bacillus strains. And this slide shows the antioxidant and antimutagenic properties of lactic acid bacteria. Based on the combination of antioxidant and DNA protective properties, we selected the strains presented on the slide for future studies. We choose strains with simulating antioxidant and antimutagenic activity. First, using control strains with known probiotic activity. In summer of 2020, we conduct a balanced experiment of feeding chickens with probiotic preparation based on the strains we isolate. In addition of the strains Bacillus subtilis KB16, Bacillus subtilis 41, and Bacillus amyloidificans 54, the strain Bacillus subtilis Catmira 1933, which has already been used in our previous studies, was used as a control strains. All supplementation were prepared by solid phase fermentation of soybeans. On the slide, you can see a table showing the growth performance of broilers in the last 2 weeks of the experiments. There is significant difference in live weights, growth rate, and feed conversation between control and chickens fed Bacillus subtilis KB41 and Bacillus amyloidificans KB54. No significant difference in gut microbiota were found. However, we found significant difference in change in the expression of interleukin 6 and interleukin 10 genes in the spleen, shown on slide. The difference were in the same groups in which there was an improvement in growth performance. We investigated the survival of probiotics, vegetative cells and spores in the intestines of bird. As can be seen from the slide, it was Bacillus subtilis KB41 and Bacillus amyloidificans KB54 that went through the full life cycle from spore to spore in the gut. This indicates that they were biologically active and capable of producing secondary metabolites. 5. Testing strains under near intestinal conditions. It is known that the biochemical activity of bacteria depends on environmental conditions. Therefore, it's desirable to study the process occurring in the intestinal microbiota, including the activity and properties of probiotic bacteria, under conditions similar to those in the intestines. Most often, studies select a significant number of potential probiotic strains. Testing them all on model animals is financially unprofitable and time-consuming. Preselection of strains under conditions close to intestinal condition is necessary. We have developed a model of atrophical chicken intestines to maintain the temperature, pH, oxygen level required for the development of the intestinal microbiota over a long period of time. This chicken goes into central tract. Development of an atrophical chicken gut medium. Due to the fact that the medium imitating the contact of intestines of bird has not been developed, we analyzed the contents of chicken's cecum for protein, carbohydrates and fats, and on this basis developed an atrophical chicken gut medium. The atrophical chicken gut medium supports the growth of intestinal bacteria, keeping the numerical ratings similar to those of the natural intestinal contents. This slide shows DNA protective and accident activity of bacillus under laboratory and atrophical chicken intense conditions. And this slide shows DNA protective and accident activity of lactobacillus under laboratory and atrophical chicken intense conditions. As you can see from the slide, the properties of the strain can change under atrophical gut medium and low oxygen conditions. This is important when evaluating how the strains will behave in the gut. future research. Another large balance experiment of chick is currently being completed. Work is also underway to evaluate the effects of probiotics, symbiotics, and biologically active substances on the microbiome of chickens under tropical intense cell conditions. In addition, we are working on the tropical gut media for the throat and dog. We are ready for collaboration and joint research on this direction. Thank you for your attention.
Dr. Mazenko, thank you for your presentation. The floor is open for questions. We have time for a couple of questions. Perhaps somebody from the distant audience. If anybody has a question, you just let us know that you're with us. And if not, anybody from our audience? No questions so far. So I have one quick question. Is that correct, based on what you are saying, that the microorganisms that perform remarkably well in laboratory condition may not necessarily perform as good in the real animal condition?
Yes, we seen some species that were very good in laboratory conditions, but in the conditions of atypical gut media in low oxygen level, they shown not very good properties.
And my guess would be that perhaps these should be included into the experiments, the process designed for the selection of the microorganisms. And last comment I have, I see the reason why you excluded the antibiotic-resistant microorganisms as candidates for these particular set of experiments, but I think it may not be a very crazy idea to keep at least a couple of them perhaps in the future, because nowadays we're talking more and more about the introduction, the early introduction of probiotics right after the antibiotic therapy. And of course we need to have probiotics that can be functional in the background of antibiotics. And there are more and more papers published that are saying that. So don't throw them away. Keep them in your collection. Maybe we'll come back to these microorganisms in the future. Thank you for your presentation.
Thank you.
We continue with our session, and I would like to give floor to Dr. Praznova.
One second. Dear colleagues, I would like to talk today about the paradigm shift that is currently underway in the field of probiotics research. Until recently, it was believed that the effects of probiotics are limited to a certain range of specific effects, let's call them classic effects. But more and more studies indicate that their effect on the host organism is more complex and systemic. Even in some yesterday's talks at this conference, the thought came up more than once when speakers talked about the effects of probiotics on the immune system or on the mental health of the host, and also the spectrum of genes that appear to be regulated by probiotics is also expanding. It seems that the new concept of a systemic action of probiotics is now emerging. The very term systemic means that the effect cannot be explained by a simple summation of many minor effects. And in most cases until recently, the action of probiotics was described exactly this way, as the summation of antagonistic or antioxidant and many other effects. And the systemic principle means an action on some critical points, regulatory points inside the organism or inside the cell. So in this concept, we are looking for regulatory cascades or key points that can be influenced by small molecules of bacterial origin. And here are some possible targets for these effects. We should keep in mind that probiotics interact with their eukaryotic hosts, but also with the rest of microbiome. In other bacteria, probiotics can influence SOS reparation, quorum sensing systems, and horizontal gene transfer systems. These processes are crucially important for bacterial cell, and there are some recent works indicating that these key points can be affected by probiotic bacteria. In eukaryotic cells, these key points are processes in mitochondria, expression of nuclear regulatory genes, for example, such as genes that triggers big cascades, for example, the P38-MAPK pathway, and the expression of genes responsible for the stress response, particularly oxidative stress, are most often affected by probiotics. In several projects of our team, we paid special attention to the anti-mutagenic effect of probiotics. We took in mind this scheme, but we didn't try to study all systemic effects. We focused on the study of the following phenomenon. A, some probiotics are able to reduce the level of antibiotic resistance mutations in other microorganisms. And B, the same probiotics appear to have the ability to prolong lifespan of the host. And both of these effects can be attributed to anti-mutagenic effects. So in works of our laboratory in Academy of Biology and Biotechnology, Southern Federal University, we conducted a set of studies. Firstly, 2 strains of Bacillus were selected, and their metabolites demonstrated the ability to reduce the expression of genes of the SOS response in Escherichia coli. The biosensor test was used, the method that Dr. Mazanko already described. The same 2 strains have shown the ability to reduce the number of antibiotic resistance mutation in gram-positive and gram-negative bacteria. And in the following experiment on chicken, which was carried out at the Volgograd poultry farm, these 2 strains were used as single preparations and as a mixture of them, group 3 on the slide. A number of biochemical and physiological effects have been obtained. The body weight gain in all experimental group was higher than in the control, and the weight of the reproductive organs of the chicken in the experimental group significantly exceeded the control. Improvements were observed in biochemical blood parameters, in the quality of sperm of the roosters, egg production of laying hens, in morphological and biochemical parameters of eggs. But the most interesting effect was the following. The inevitable decrease in egg production in laying hens in group 1 occurred more slowly than in the rest. The slide shows the relative egg production calculated as a function relative to the control. A fraction with and also a preparation based on Bacillus subtilis catmira strain that also was mentioned today caused a decrease in the number of mitochondrial DNA damage by 34% compared to the control. A fraction with SOS inhibitory activity was isolated, and an oligopeptide fraction with thermal stability and a mass of less than 3 kilodaltons was isolated by mass spectrometric analysis. These properties correspond to the spectrum of non-ribosomal peptides secreted by the genus Bacillus representatives. Bioinformatic analysis of the Bacillus subtilis strain genome showed that this strain is capable of synthesis of 4 non-ribosomal peptides that are presented on this slide. The mass of them corresponded to the peak found by mass spectrometry. It is a surfactant-like molecule. Surfactant is known as an antifungal compound as well as a quorum sensing regulator. It should be noted that some antimicrobial molecules can have the additional function that can manifest in subinhibitory concentrations. In particular, bacteriocins have a dual action sometimes and can also be used by microorganisms as signaling molecules at subinhibitory concentrations. We think that this surfactant-like molecule can be the intermediate of the above-mentioned effects. In conclusion, I would like to highlight these key points: that probiotics have systemic effects on host. Some of these systemic effects are closely related to antimutagenic properties, and antimutagenic effect expresses in microbiota, in both microbiota and in host mitochondria. The fraction of metabolites responsible for this effect contains surfactant-like molecules, and the mechanisms of their action is still to be discovered. That's all from me. Thank you for your attention.
Thank you for the most interesting presentation. And before I will attack you with my questions, I want to ask if there are any questions from the audience, and can we check the distant participants? If anybody has questions to the presenter, please let us know. Anybody from the audience here or the distant audience? I can see that Rivaz is still with us, Santosh is still with us. If you guys have any questions, just let us know. But in the meantime, I will start. I have a couple of questions. One of them is more philosophical, if you wish, and another one is more practical. The philosophical question. What really stroked my attention from the first slide of your presentation was the qualitatively different approach to probiotics with the systemic effects. It's a very different and more in-depth approach as compared to the one that was used and still used, but was established historically the first. With systemic approach, do we have to consider some perhaps different tools or different lines of selection for the potential probiotic? What is your take on that?
Yeah, thank you for the question. I think that, and it's not my personal opinion, it's an opinion of of all my colleagues, that on the stage of primary screening, we need to select probiotics with antimutagenic properties or antioxidant with such properties that can be the markers of such systemic activities.
Thank you. So to generalize perhaps even more, I think it will be appropriate to say that this set of biological markers should be established, which will be a ground for the selection. Because when we look at the definition, the current definition of probiotics, the most important thing, well, health benefit, but of course health benefit comes with specific biological markers.
Yes, and also I think we need some test systems based on mitochondria effects effects on mitochondria.
Yes, thank you. And the second more practical question I have, the more practical question would be, you mentioned surfactants in Cadmira 1933, and I know that the genetic analyses also revealed possible presence of the gene coding for nattokinase. So we've never tested it, actually. And now, being in a bigger audience with many other participants here and virtually from all over the world, if anybody knows about a quick and easy test for nattokinase, please share with us, because we really want to see if the genes are really coding for this very interesting molecule, which is also known as a health-promoting molecule. So genes for nattokinase are in Cadmira 1933, but we don't know if this enzyme is produced.
Yes, we don't know if they are expressed.
Yes, so that's one of the things, one of the challenges we have in front of us. And we have time for one or two. Yes, Santosh, please.
Really, this is a very interesting talk, and is a very good approach by using systemic techniques. So, my interest is at this time, the probiotics have a kind of multifunctional approach, as we know already, and this approach will solve this purpose, I think, to the maximum extent. And what about this immune modulation study, like systemic approach if you are doing, can we understand up to what extent the immune modulation is there? What technique you can use? What I would like to know secondly, if any bioinformatics approach before moving to systemic will be more beneficial to understand. What is your feeling?
I didn't quite get to the last part of the question.
Perhaps I can try to help. So Dr. Kulari was talking about the perhaps good direction using systemic approach, but he's also curious, and Santosh, please correct if I'm wrong, your question was about how we can also have in this systemic approach the immunomodulatory function being selected at the level of biomarkers, and perhaps the information about the genome, if it can also help in understanding whether the microorganism is potentially in the group of putative probiotics. Is that correct? That's what you wanted to ask.
Yes. Yeah, you are right.
Thank you for the question. It's a very promising direction to the further research. And I think thinking about test systems, we need to establish the mechanisms of immunomodulation first. So yesterday I've heard information from some of talks that there are two proteins that are able to communicate with the immune system of the host. That's proteins that's secreted by bacteria and communicate with the immune system of the host. So if we know the exact proteins, we should use their genes as markers in testing, in primary testing for screening for probiotics.
Thank you very much. If there are no more questions, yes, Dr. Laptev.
Maybe it is my misunderstanding. Do you use any mutagen creating the intestinal microflora?
But this is a good question, whether the mutagens are used or not, and the short answer is no. However, nowadays, more and more we see... there is one more thing. The laboratory evolution is the answer, and that's an approach that is becoming very popular. proposing the mutagenicity. If we don't have any more questions, we're moving. Thank you very much for your presentation. We're moving to the next presenter.
I just wanted to answer the last question. The short answer is we didn't use mutagens, we used anti-mutagens.
Yeah. So the next presenter is Dr. Isaeva. The floor is yours.
Yes, I'm here.
We can hear you.
Yes. Yes. Так. One second. Okay. Okay. Hello, dear colleagues. I am from Kamchatka State Technical University, Department of Aquatic Research, Fisheries and Aquaculture. Me and my student Nina Kristina show for you presentation using natural aquatic organism as a taste attractants for aquaculture of salmon. Our purpose and objective of our study was development and additivities and technology of the user waste obtained during cutting of marine aquatic organism for the production of feed additivities. We are studying the taste preferences of salmonids for various types of food and chemical stimulus, to study the palatability of artificial feeds used for rearing salmonid juveniles, to objective to palatability of marine raw materials waste. To develop a technology and introduction food extract into pellet feed. Our material collected up to 2 years. The first we are cutting this net fishing near the upper rivers to Paratunka River chair and Karimaiskaya channel of the Большая Быстрая River, Koha Salmon. We are watching the experimental setup and aquariums volume of 5 liters. On April 51, we are catching 200 juveniles of cham salmon. We are cutting in salmon farm in the village to Ketkina and devalued to the laboratory under similar conditions. It's our laboratory, it's a farm. Experiments were performed using methods, behavioral test reactions to artificial fish feed pellets soaked in the aquatic extract hydrobionts. Cellinus, it's 20 copies Koha salmon and 60 specimen cham salmon. Sized and given in the table. The number of experiments carried out on chair was 838 black salmon to other. These Ancharinkus kizuch, Salvelinus malma, and Ancharinkus keta. Research methodology. Water extract invertebrates, shrimp, bloodworm, larva, apelio crabs, is artificial feed, company AgraServer, statin feed. it's a conservation in this concentration. Using flour substance were introduced into the composition in the granules. Regular feed pellets were used it as control pellets. We used in this mission first aquatic organisms were vegged and kided in mortar and gradually mixed and set at water until homogeneous. The resulting extract stored and refrigerated furthermore, and then one day. Dry granulated food pellets was soaked in aquatic extract of the extractant for 1 hour before feeding the fish. Result, among the granules with extract of marine organism and alga, crab, shrimp, bloodworm, larva, cats, must of tested object has the attractivity test for extract head in different property and the deterrent properties were found for juvenile coho salmon. Granulated containing shrimp extract is a concentration 30 gram 2 liters, had a maximum palatability for juvenile coho salmon. This picture showed the different extract and how palatability pellets. The result for juvenile chair. The most activity for chair were granulated containing the water extract of shrimp. Their consumption for 68 up to 9% and comparison with the control granules. The granules containing the bloodworm extract were slightly less attractivity, 65, but 6%. Consumption of granulated and artificial feed extract was the level of consumption control granules and did not differ statistically. The shrimp pellets was readily consumed immediately after single set, and contrast the after pellets where several grabs were noted in the experiment. In the result for juvenile cham salmon, the master activity stimulus for cham salmon is snow crab extract. Cham salmon reacted the reaction theta very activity and feed this additivity more actively than to dry granulated compound feed. The reaction cham salmon to granules with extract manifested itself instantly as soon as the granulated enters the water. Young cham salmon feed more activity in the morning before lunchtime. It shows the juvenile cham salmon activity eat pellet food, snow crab attractants. Conclusion. It's the result of the experiment is found the used extract of aquatic organism have a highly attractivity test for coho salmon and chum salmon and expect for granulated with bloodworm extract and and different stimulus that the consumption did not differ significantly from the consumption of drew food granules. The most activity appeared in flowering stimulus with shrimp and snow crab extract pellets for concentration using this significantly increased pellet consumption for up to 98%. The result obtained showed with natural food items, marine aquatic organism and alga can be used as attractants and enhance the consumption of irrigation activities at artificial pellet food. The work concurred of the laboratory in the Department Aquatic Bioresearch, Fisheries and Bioresources, Fisheries and Aquaculture with the financial support to the Russian Fisheries Agency. Thank you very much for your attention.
Thank you for the very interesting presentation. We have time for a couple of questions, and I believe the presenter is with us right now. Any questions from the audience? Yes, please.
Dialgo, thank you for talking. Do you research physiological parameters?
Yes, we research physiological parameters and histological parameters in fish, but it's not published data. We are working.
Thank you.
Now.
So we understand it's still a work in progress. It's an ongoing study, right?
Yes, yes.
Okay, thank you very much for your presentation.
Thank you very much.
Thank you, and we're moving to the next presenter. I believe we have now Dr. Abayimova. Not doctor yet. Not doctor yet. Junior researcher. Yes. Right. Okay, so all the beauty of the growth, professional growth is still in front of you. So the floor is yours.
Good day, dear colleagues. Allow me to introduce myself. My name is Alena Abayimova. I'm a junior researcher of State Research Center for Applied Macrobiology and Biotechnology, Department of Molecular Macrobiology, Antimicrobials Laboratory. So, in the search of new alternatives to antibiotics, scientists' skeptical attention turned back to bacteriocins. Yesterday, Professor Deep from Norway already gave us a small review to the nature of bacteriocins and classification of bacteriocins of gram-positive bacteria. A classification that was first made in 1993 by Mr. Klenhammer and constantly changing ever since due to the great diversity of bacteriocins. There were 3 classes, and 4s, and 5s, and already now it's again 3. Information constantly updating, subclasses dividing, integrating, renaming, and so on. But our subject of interest today is more stable. Mundicin E28 belongs to subclass 2A, unmodified pediocin-like bacteriocins, probably one and only subclass that wasn't renamed or modified. Mundicin E28 produced by Enterococcus mundi, E28 isolated from sour milk by us in 2008. Mass spectrometry analysis of active fraction by Malditov-Brucker, PCR analysis, genome sequencing showed that mundicin E28 is identical to previously described mundicins such as mundicin KS, described by Kawamoto and Sonomoto, and mundicin serial 35, described by Farias. The active fraction of mundicin with a purity about 70% was obtained by a one-step elution from sorbent Cefadex CM25 that excludes any peptides larger than 25 kilodaltons. And the fraction with a purity about 90% was obtained by reverse phase chromatography. The antibiotical activity remained stable due to a wide range of temperature even autoclaving was remained pH stable, enzyme stable, except proteinase K and alpha chymotrypsin, and remained stable after lyophilization in different protective media more than a year. The antimicrobial spectrum of mundicin E28 includes such human pathogen as Listeria species, Enterococcus species, Clostridium perfringens, and several other strains of foodborne pathogen species, which not shown here because there were only several. With a MIC in nanogram range that's significantly lower than antibiotics. All of these pathogens also are foodborne pathogens, so-called, and among them, our point of interest is Listeria monocytogenes, the cause of listeriosis. The ranks third in the most mortality among foodborne bacterial pathogens, and rate with Salmonella and Clostridium botulinum. The main factors in transmission of infection are food products, including poultry products. And that we have a target, we have an agent, we have a MIC for that, but simply having MIC is not enough. In practice, in practical application, always everything always works different than when you're working in the lab. So we monitoring several poultry processing in slaughterhouses, so different poultry farms, and found several thank you. And found several points of high risk of contamination. Listeria monocytogenes, its feather and legs, cooling baths, and semi-products in boxes. And develop the places when we can actually apply to mundicin treatment, such as cooling bath and yes, semi-products in boxes. The first step and the second step was simulate these critical points, and for that we used Listeria contamination model as short-term immersion in a suspension of test strain Listeria monocytogenes ATCC19111 in concentration on 10⁶ CFU per milliliter. And after bacteriocin treatment of samples, there always was washing from the samples were titrated with serial dilutions with spreading of sample of each dilution on PAL agar in duplicate to reveal the Listeria contamination. So the first we decided to start with a simple. They tried to decontaminate the water from Listeria in amount a little bit larger than in MIC volumes. It was about 100 milliliter, and here we used the concentration of mundicin E28 and like in 2 orders of magnitude larger than MIC range for Listeria monocytogenes ATCC19111. It was 2 nanogram per milliliter. And we found that actually decrease by 5 orders of magnitude was in 40 nanogram of milliliter concentration, not even 20. And it was remained stable over 6 hours, only in 24 hours after a day of exposition, the Listeria start to grow again. And based on that, we tried to simulate of cooling bath, and we used a concentration of bacteriocin 14 nanogram per milliliter, and there were 4 broiler carcasses, which was previously washed with water to determine the initial contamination, possible contamination. After that, they were short-term immersed in suspension of our test strain of Listeria monocytogenes. 2 of them were contamination control or positive control, and 2 of them were short-term immersed in bacteriocin solution and dried and washed off for the dilutions. It's almost like in some Russian fairy tale. And there were 2 points of exposition. First was made in 30 minutes, and 2 broiler carcasses, 1 positive control and 1 treated, was left in refrigerator for 1 day, for 24 hours. And we found that also Listeria contamination decreased by 3 points of orders of magnitude and remained stable during the day, during the 24-hour. The next, we try to simulate of cooling nozzles, because in different farms it's sometimes used instead of cooling baths, but works with broiler carcasses actually not very comfortable in the lab conditions. So we, instead of that, we used broiler wings that were bought in supermarket and contaminated the same way, but they were dried and sprayed with mudicin 28 solution, in different concentration again. And again, we found that in concentration 40 nanograms per milliliter, Listeria contamination decreased by 3 points of orders of magnitude, and at concentration of 160 nanogram per milliliter, there was a complete elimination of Listeria from the samples in both temperatures. But firstly, we show on Palagar some growth, typical Listerial colonies in negative control. But by typing by mass spectrometry MALDI-TOF, it was confirmed that isolates are Listeria innocua, not the Listeria monocytogenes that we used for contamination. That showed the natural Listeria contamination of poultry products in our supermarkets and retail network. And the last but few stages step, we decided to discover the biopreservative potential of mundicin E28 in biopreservative of semi-products. For that, at first we used cling film that what sorbed the mundicin in concentration of 40 nanograms per milliliter. And yes, in 4 degrees of Celsius, it was shown the same progression. And also we use the step with simply sprayed mundicin as before that. And it sometimes somehow shows a better result than the cling film with sorbed on mundicin on it. Okay, it was not what we wanted to see, but still that's the result. And at room temperature, sadly, wasn't possible to check the result because of great massive bacilli growth. So we cannot count the Listeria contamination results. And as a second stage, we tried to use cling film with absorbed actually alginate monolayer with absorbed of mundicin, as our colleagues in other studies, for example, for combine their bacteriocins for treatment Clostridium, anti-Clostridium activity. And yes, it shows completely different results, but they seems good, but strange for me at least. And they also were some unspecific not Listeria growth at 25 degrees of Celsius at room temperature. So The next point, we tried to research the decontamination of minced chicken with the possibility to extend its shelf life also as a biopreservative. And that time we used two concentration. First is very known 40 nanograms per milliliter, is there a mistake? And the second time is 160 nanograms per milliliter to see is it will eliminate any unspecific grow, so we can check the results. And there is still a decrease in Listeria contamination by 5 of the orders of magnitude, and almost complete elimination at concentration by 160 nanogram per milliliter. And at room temperature, the samples actually spoiled at 4 days, so it's not very extended. But when we count the total viable count, there were a significant amounts contamination this minced chicken. So we actually hardly can count the number of colonies. There were actually on TTC agar, for example, like 10 or more types of different colonies, so different species. That leaves room for questions of quality, the poultry products in our network. Let me make a conclusion. The Listeria contamination to treatment of Mundocin E28 decreased 5 orders of magnitude at 4 degrees of Celsius and 3 orders of magnitude at 25 degrees of Celsius in 40 minutes, and remained this level for 3 days and more at a concentration of 40 nanograms per milliliter. And at concentration of 160 nanograms per milliliter, a complete elimination of Listeria from the test samples was observed. This is a large amount, but still it's nanogram range, so it's lower than usually antibiotic range. So we can recommend Mundocin E28 as an anti-listerial agent in slaughterhouses of poultry farms, and as a potential biopreservative for semi-products, but still our models sadly require some more tests and maybe development, so we can get more appropriate results. Thank you for your attention.
Thank you for your very interesting presentation. I'm asking our colleagues who are with us today here in the audience personally and distantly. Are there any questions to the presenter? I will start asking my questions, and we'll see if there will be other. So we have time for a couple of questions. This is a class 2A bacteriacin. Yes. I wonder if you compare, and you mentioned that it's homologous, identical to another bacteriacin, which was perhaps isolated by another group.
Yes, it was identical to other previously described Mundocin, except Mundocin L, nearly described, because there are lysine instead of valine in structure, but other previously described Mundocins are identical.
Okay.
But
so yours is slightly different. Question, did you compare your class 2A bacteriacin to any other in regard to activity against Listeria?
Yes, we compare, actually, we check the results. That's why we actually research wide spectrum, antimicrobial spectrum activity, because other authors took also Listeria species, Enterococcus species, Clostridium species, but mostly test strains. And we also check the activity on field strange isolated from patients, from those who die, from the environment. That was on slide with the MIC ranges. There were also a range in MIC, and sometimes great range, nanomolar concentration, but still range.
So what we see is that yours has a broader range and perhaps is more active as compared to...
I allow myself to correct you. Our more broader range. It's just the investigation that we made instead of that authors, because we wanted to check also foodborne pathogens for biopreservative perspective.
Understood. Thank you. And well, the drop in the viable cell count, 3 logs, is a good one. Also considering that this is not a ready-to-eat product, so the thermal processing should, precede the consumption of the product. I was also following with interest your study on the control of the microorganisms from the film. With the alginate, I believe one of them was alginate, and the first one, which wasn't that successful. How did you apply your bacteriocin to the film?
To the film?
To the plastic, yeah.
Yes, it was also immersed in bacteriocin solution and sorb on that, and then dried. And we checked the activity. Yes, it was active. So the sorption process took the place.
So basically, there may be a challenge, and the challenge comes with the rate of transfer from one solid surface to another solid surface. So you need to have some bedding. So alginate creates this opportunity. That's number one. Number two, you perhaps I don't know, maybe you already studied it, or you should study the diffusion rate, and to see, what if it follows Fickian diffusion or is it non-Fickian diffusion, and that is also very important for the activity, because then you will know how much of the bacteriocin is coming into the target at each moment of time. And that will certainly help to improve. And the last, not a question but suggestion, you may consider using more than one natural preservative, I don't know, like weak organic acid or something, because there is a good chance that they can work synergistically and the overall effect can be extended. You may consider,
Yes, we definitely consider it. Thank you. Thank you.
Okay. Other questions to the presenter? I'm asking our audience, both distant and local. If no questions, thank you very much, and we'll move with our presentations. There was a challenge how to arrange the next one, and I think we finally have the solution. The next presentation will be by Dr. Ushakova, Complex Bioactive Supplements for Aquaculture, and I'm asking our assistants to start the presentation.
Dear colleagues, let me introduce our report, Complex Bioactive Supplements for Aquaculture. As an alternative to antibiotics, in the last 3 decades, special attention has been paid to the dietary administration for biologically activity additives with the feed. The application of therapeutic and feed additives in the aquatic environment has its own limitation compared to terrestrial condition, as washing out and dissolution of active components of the feed granules may take place in the aquatic environment. Therefore, the strategies that are being applied in this area and those related to aquaculture are for particular interests. It's generally accepted that probiotics, prebiotics, metabiotics, and medicinal plants in the form of phytobiotics are the most promising feed additives for the control or treatment of various diseases of aquaculture objects. Evolutionary, probiotics were developed as the first feed formulation for prophylactic purposes. Probiotics are lived microbial additives to feed used in fish cultivation and farming, as well as the cultivation of crustacean and mollusks, as an effective and environmentally friendly alternative to antibiotics. That is, the action of probiotic is isolated with a protective strategy as an alternative to antibiotics in aquaculture. In addition, positive effects have been obtained when using probiotics against viral and parasitic disease in aquaculture animals. The use of bacilli probiotics in aquaculture improves feed utilization, reduces stress responses, increases immunity and disease resistance. A prebiotic contains components that mechanically cleanse the intestinal walls, have anti-inflammatory effects, improve motor skills and peristaltics, normalize carbohydrate and fat metabolism, bind and detoxify. The effect of dietary supplements, then essential oil of Thymus vulgaris, on the allochthonous microbial composition of rainbow trout was evaluated. Metabiotics are defined as a structure component and biological active metabolites of probiotic bacteria. Metabiotic formulation, as a rule, do not contain valuable cells of probiotic bacteria. With metabiotic, it becomes possible to establish a controlled intestinal microbiota that is not dependent on the quantity or distribution of microbial species in the environment. Active metabolites have a complex of positive effects. Antibacterial properties allow you to fight pathogenic and opportunistic microorganisms without affecting the beneficial intestinal microflora due to the enzymatic activity or hydrolytic enzymes. Digestion is improved, the body's immune defense is enhanced. In recent years, many scientists working in the field of animal nutrition have increasingly focused on the use of phytobiotics, which are defined as biological activity substances formed in plants. Phytobiotics are readily available for use as component of animal feeds, where they may be added for the antimicrobial, anti-inflammatory, antioxidant, and antiparasitics. Quorum-sensing inhibitory compounds have been isolated from many different natural sources, including traditional medicinal plants, herbs, fruits, and vegetables. All the positive properties of the least substance can be combined into a single complex biotic. The prospects are opening in connection with the use of complex biotic obtained by solid phase fermentation of phytocomponents by probiotic bacteria. Under condition of bacterial cultivation on solid cellulose particles with reduced humidity and limited access of oxygen, similar to the condition of natural vital activity of the intestinal biota, where cells of the probiotic culture from a biofilm and secrete various metabolites, including organic acid, enzymes, and signaling molecules. Solid phase fermentation of plant substance is used to obtain various biologically active substances which enrich phyto-raw material with value product of bacterial destruction of the substrates and specific metabolites of probiotic. On particular interest is the substrate fermentation using Bacillus subtilis. For example, cell extract and cell-free supernatants of Bacillus subtilis V8130 have been shown to contain somatostatin-like peptides. It has been shown the solid phase anaerobic fermentation of fiber-containing plant substrates such as cibactone meal, sunflower meal, beet pulp, which is carried out by Bacillus subtilis V8130, associated with biochemical transformation of plant polymers by vegetative cells on the bacillus. In the composition of the fermentation product of beet pulp, various substances were found, many of which have bactericidal activity, various phenolic compounds, and their derivatives. The presence of those substances in a complex probiotic formulation undoubtedly affects its biological activity and is of great value. Among the secondary products of bacilli metabolism under condition of solid phase fermentation of the plant substrate, attention is drawn to a compound the most probable structure of which is 5-heptyl-2-dihydrofuranone, and a substance having a structure similar to the A factor of same actinamide sets containing the gamma-butyrolactone cycle. The specialate that the mentioned metabolite of Bacillus subtilis V8130 is able to act similar to the A factor of actinamide sets, is a regulator of secondary metabolism and participate in the establishment of a biofilm and phytocarrier and maintain a symbiotic relationship between a microorganism and a host. The Bacillus subtilis vegetative cells biofilm is shown in the slide. When dry, the cells are protected by the biofilm network. Probiotic formulation balanced by phytocomponents are distinguished by increased biological activity due to the combination of the probiotic effect itself with the presence of metabolites as well as the action of biologically active components on the phytobiotic. The microbial quorum sensing is likely to play an essential role in intracellular communication and survival of vegetative cells of multi-microbial probiotic prostor, consisting of the products of solid phase fermentation of Bacillus subtilis V8130, Bacillus subtilis 29, 84 and Bacillus subtilis 4099 grown on the beet pulp. Formulation prostor is distinguished by the additional inclusion of herbal phytoparticles, milk thistle, Silybum marinarum and Echinacea purpurea. Echinacea has immunomodulatory properties. It's proposed to introduce Echinacea purpurea into the phytobiotic prostor is to improve the immunity of aquaculture objects. Phenol hyperxylic acid contained in the root of Echinacea station the immune system have antibacterial, antiviral, antifungal effects. Milk thistle is used to prevent various liver damage, improve a digestive process. Milk thistle is unique in that it helps cleanse and form new liver cells. The introduction of medicinal plants derived into phytoparticles into prostor allowed the formulation and production of a complex biologically active feed additive containing probiotics and medicinal herbs, targets for animals, poultry and fish. The modification of the prostor formulation is the Herbastor products contain the yeast autolysate. Amino acid nucleotides, amines, monosaccharides and autolysates vitamin have an additional sources of nutrition and probiotic bacteria. Yeast cells will contain manana oligosaccharides and beta-glucans, effectively bind and absorb various pathogenic microorganisms in the gastrointestinal tract, including Escherichia coli, Cladostrum, Vibrio, Salmonella, thus reducing the possibility of infection. In addition, beta-glucans have a stimulating and optimizing effects on the immune system. Complex biotic Herbastor is a product that represents a new class of bioformulation superior and efficiency to those on the market. The introduction of medicinal herbal imparts additional biological properties on this formulation. That's the biofilm of the raw Herbastor complex with live cells of probiotic bacillus and inactivate yeast and on a complex phytocarrier. The stimulators use the medicinal plants and probiotics is not limited to their synergic action. Formulation of complex biotic increase digestion efficiency, feed conversion, grow rate, optimized aquaculture productivity. The case of carp and sturgeon species juveniles conducted previously provides the efficiency of the prostor preparation of fish species. Formulation in the amount of 1.5 kg per ton of compound feed was introduced into the commercial compound feed of carp, and in the amount of 2 kg per ton of the compound feed for sturgeon. Under yearlings were kept in aquariums in group of 15 individuals. The fish breeding and biological indicator of juvenile carp and sturgeon fish in terms of absolute gain and average daily growth rate were higher compared to the control for carp, respectively by 45% and 25%, and for sturgeon by 120% and 45%. In sturgeon fish in the experiment, the survival rate of juveniles was 13% higher than that for the control fish. The experiment with growing sturgeon juveniles, hybrid of the Russian Lena sturgeon, on a diet with a modified complex biotic herbastor in an amount of 0.1% in a production compound feed for sturgeon fish in close water supply units, the absolute and average daily grain significantly increased. You can see this on the slide. The access to the physiological state of the fish and analysis of the hematological parameters of the Russian Lena sturgeon hybrids was carried out. According to these parameters, the physiological state of the hybrids was within the normal range. A stressful situation for farm-raised fish can be confirmed by increased level of cholesterol in the blood of the fish, both in control and the experimental population. In wild sturgeon, cholesterol levels rarely exceeds 1.3 to 2.9 mmol per liter. In experimental fish, it did not exceed 2.4 mmol per liter. The concentration of total lipids during the study period on the experimental fish slightly exceeded the norm, 3 to 5 grams per liter. This allows us to conclude that the direction of fat metabolism in this fish contribute to the normal accumulation of energy resources. As a result of evaluating the effectiveness of introducing a complex formulation into the composition of the production sturgeon compound feeds, an increase in the productivity of growing Russian Lena sturgeon hybrids in industrial conditions was established. The need to increase the resistance of all aquaculture objects to infection and invasive disease, increase the growth rate, increase the efficiency of feeding, environmental safety and safety of product intended for human consumption stimulates their development, and use of various biologically active additives in aquaculture, both based on probiotics and using medicinal herbs. Thank you for your attention.
Thank you for the presentation. Dr. Ushakova asked all the questions to be addressed to our organizing committee by email, and then we will be able to answer, we'll be able to pass the questions to Dr. Ushakova, and she will be able to answer them. With these, we finish our morning session on animal probiotics. We're having now a coffee break, and we will then return back at 11:40 to start the the last session on the probiotics biotechnology. Thank you all, and those of you who are with us distantly, please come back in, what is it, like half an hour or so to start our next session. Thank you for your attention.
north or south, east or west, angels or demons. The philosophy always tries to frame everything in the bipolar model and to explain processes, framing them in the convenience life ideas. Are microorganisms read our philosophy books? Are they following our ideas for good or bad? Or are they just behaving according to natural physiological life processes? Convenience and natural processes of biochemical reactions and basic of life science processes are more relevant in microbial life compared to the human society physiological theories. So how we can try to frame our bacteria to see if they are good or bad? Are they really good or bad, or they behave just because they wanted to be work like that? In our work, we're always trying to find the microorganisms they can cover different beneficial properties. We're looking for biopreservation properties, we're looking for probiotic properties, we're looking for technological properties. But it's really possible to have 3 in 1, are really possible to have one bacteria. They can cover all these exigencies and to serve as a beneficial bacteria. This is the question. So we try to work and to try to isolate the different bacterial species in order to develop a nice technologically relevant culture that will have the beneficial properties. They will be good as probiotic. They can even have the good biopreservative properties. So we screened the different dairy products from northeast of Brazil, different milk samples, different cheese samples, and we isolated a large number of bacterial cultures. We differentiated them, and we do the appropriate classification based on 16S RNA sequencing. On the other side, we wanted to see if these strains have the probiotic properties, if they're producing the bacteriocin, but as well we needed to check their safety assessment with the idea to develop the new starter cultures. In the first step of our work, we had quite a good collection of promising bacterial isolates that was showing a very interesting antimicrobial properties. So we wanted to show that these strains are different, they're unique strains, and they as well needed to do their identification. For this purpose, we performed the RHPD PCR with different primers, as well REP PCR with the proprietary primer. And we selected, based on this screening process, 2 isolates for our future study. They showed that was actually different strains. These 2 strains, named K14 and K51, been identified based on the 16S as Streptococcus infantarius subspecies infantarius. When we talk about the different Streptococcus strain, we need to keep in mind that it's a very diverse group of lactic acid bacteria belong to the group of Streptococci. And majority of them actually it's a very well-known pathogenic species. Only few of them are beneficial, and everyone's supposed to know Streptococcus thermophilus, one of the strains that is used for the preparation of the yogurt together with Lactobacillus delbrueckii, subspecies bulgaricus. However, the question is Streptococcus infantarius a safe or not safe strain? The literature showing that this strain been isolated from the fermented milk product from West Africa, and there is a predominant species in these dairy products. Colleagues from Switzerland was studying very deeply some isolates of species Streptococcus infantarius or species infantarius, and it's really questionable if these strains belong to this species are safe or not. According to the bulletin of the International Dairy Federation, only 4 species belonging to the group of Streptococcus could be considered as a safe, and this is Streptococcus galolyticus subspecies macedonicus, Streptococcus salivarius subspecies salivarius, Streptococcus salivarius subspecies thermophilus, and Streptococcus thermophilus. And unfortunately, Streptococcus infantarius is not in this list of this recommendation. So the question is, Streptococcus infantarius safe or not? So we found this strain predominant in the dairy products in the northeast of Brazil. However, is this strain really good or bad? As first step of our evaluation, we wanted to see what kind of aggregation properties have these two strains of Streptococcus infantaris. So we evaluate their autoaggregation and their co-aggregation properties together with some strains of Escherichia coli, Listeria monocytogenes, Salmonella diffumirum, and Clostridium perfringens, some well-known pathogenic strains. It was interesting to observe that two strains of Streptococcus infantaris can show quite high co-aggregation properties with these four pathogens. But is this good or bad? Is this really can make us happy and think that these properties, high co-aggregation properties, really will be a positive effect for our strains or bad? So we needed to go further. We wanted to evaluate how will be their proteolytic activity and hydrophobicity. So you can observe that two strains have the quite different behavior related to their hydrophobicity and proteolytic activity. Much more high hydrophobicity was observed for the strain 51 compared to K14. As well, similar results was observed in the proteolytic activity, very high proteolytic activity for Streptococcus infantaris K51 compared to very low activity for the strain K14. Same time, when we evaluated survival rate of these two strains in the model system, modeling our gastrointestinal system, we observed that they have quite good survival, but again, much more better survival for the strain 51 compared to the strain 14. So all these results was showing that these two strains can survive very well in our gut. They can maybe having a good adherence inside our gut. They can make a good agglomeration, good autoaggregation with several pathogens. And taking into consideration that these two strains were showing a very good antimicrobial activity against several pathogens, most probably we can consider that they will be a good idea that these two Streptococcus infantaris strains maybe will be able to reduce the level of some pathogens. However, not everything is black and white, because we found that some of the other results for these strains in fact can be maybe confusing and maybe can be showing that a negative part of these two strains. In addition, these two strains were showing that they can carry genes for the folate production. So two strains of Streptococcus infantaris could be a very good producer of vitamin 9. Moreover, PCR analysis of the DNA obtained for these two Streptococcus infantaris strains showed that these two strains can carry several genes related to virulence of these strains. The two strains was showing that they can have the gelatinase activity. They can carry the gene involved in the enterococcal surface protein, endocarditis antigen, addition to a collagen, even vancomycin resistant of vancomycin B, and histidine decarboxylase. All these virulence genes was showing that these two strains cannot be considered as beneficial because they was carrying 6 from 13 tested genes. So a very high level of the virulence for these two genes. In addition, some specific streptococcal virulence genes was showing a presence of extracellular factor. So obviously, based on their genetic heritage, these two strains cannot be considered as In addition, two Streptococcus infantaris strains were showing production of the bacteriocins, antimicrobial peptides, as well as based on the cell-cell interaction, was showing that can inhibit several other organisms. From one side, inhibition of Salmonella diffumirum or inhibition of Staphylococcus aureus could be a good idea, because showing that these two strains can eliminate from the system some pathogens. Escherichia coli as well was showing to be sensitive in the cell-cell interaction, as well some of the Clostridium perfringens. However, bacteriocin activity against several lactic acid bacteria was a fact, so bacteriocin produced by these two Streptococcus infantaris strains will be able to inhibit several of beneficial organisms. So again, it's putting the question, is this good or bad? Moreover, we saw that these two strains, based on the physiological And when we perform the antibiotics resistance for these two strains, we observed that these two strains can be resistant to a very big list of different antibiotics. So putting all these arguments together, from one side, we have a strain that is very good survival in the gut. They can be very well adhere to our gastrointestinal part. Strains, they will be able to eliminate several of other organisms, including several beneficial organisms. So from this point of view, these two strains have the good chance to survive in our gut. They are lactic acid bacteria, they are streptococcus, but streptococcus that are not considered as a safe. And from another point of view, they carry several virulence factors. Thinking a little bit further, if we have these strains in the dairy production system, they will be able to eliminate the other strains from the system because of their good competition ability to compete with the other organisms, not only pathogens, but as well the beneficial organisms, and they will be taking all over in the ecological system. Moreover, because of their good survival with the gut, they will be able to install and feel very comfortable in our gut. So in fact, these strains can be considered like a very dangerous because of their virulence factor they can carry and their good survival in our gut. So bottom line will be that Streptococcus infantarius K51 present a better potential to colonize and compete with the gut microbiota compared to Streptococcus infantarius K14. Both strains present resistance to the major group of antibiotics, and they can carry different virulence factors, so make them a potential biological hazard for the humans and other animals. And milk containing these two strains is a health risk, since the study of Streptococcus infantarius strains presents a good ability to survive in stimulated gut condition and will be responsible for the some chronic gut disorder because of the presence of the different virulence genes and antibiotic resistance. When we're talking about the lactic acid bacteria and bacteriocins, we need to think a little bit more further, because in the past we was talking that the bacteriocins is only natural killers. They can kill the other organisms, normally closely related. However, in the literature, it's more and more emphasis is given to another fact, the fact that bacteriocins can play much more roles in our cell life, in our microorganism ecosystem, because of their signaling properties. Some of the bacteriocins can be involved in the current sensing process. They can be involved in some interaction with some eukaryotic cells. Even some authors were showing that can have even anti-cancer activity, spermatozoa activity. They can even be involved in the control of some viruses. So I want to thank you very much for your attention for this presentation. Большое спасибо. And I want to thank to all my collaborators from all around the world. From Asia, it's Handong Global University, several universities from Europe, Stellenbosch University from South Africa, and several universities from Latin America. And collaboration, in fact, with all these colleagues was making this work possible. And for the end, gentlemen, it is the microbes who will have the last word. Thank you very much. Большое спасибо. I would like to thank you very much for your attention. And if you have any questions, I will try to answer.
Can I try, Slavi?
Yeah, I'm in trouble.
Okay. Good afternoon, Slavi. Good morning to everyone. I'm Jon from Norway. I'm wondering how you genome sequenced your two strains to see whether they have genes involved in competence or not, since you have a lot of resistant genes in your settings or in your experiment?
Actually, we found that these genes, these two strains, cannot be considered as a safe. Our original idea was to use them as a starter culture.
Yeah.
Because they was having extremely good survival, but after we start to going deeper and deeper, we discovered that they actually quite very bad guys. And they was isolated several times after that from the same area that you isolated the first time. So I mean, they're very stable. On another side, there is a couple of papers coming from the people from colleagues from Switzerland, evaluating the different dairy products from Eastern Africa. And they're showing that these Infantarius could be predominant strains in the several Eastern African dairy products. So look like they're quite popular. So we, from one side, we saw that they're bad guys, so we don't want them. But from another side, we have this scientific curiosity to see what is going. Yes, we sequenced one of them, strain 1.5, and we found that it's carrying not only the several more even virulence factors, but as well they are able to express even more virulence factors. They're able to compete very well because they had several addition genes involved in their genome. So they will be really able to survive very well in all the environment, not only in the dairy environment, but as well in our gut. So, but again, this was a curiosity, but since there was not beneficial, there was a bad guys, we was a little bit step out of them. But this is, I think it's a good example because in the last couple of years, they tried to rehabilitate some of the streptococci, not only Streptococcus thermophilus to be considered as a good player, but again, we need a really deep study before we rehabilitate any Streptococcus, because we can enter in the very dangerous problem. Because if we look only on the beneficial properties of some strains, we can see they have the beneficial properties. But as well, we need to look on the dark side, because before we introduce them to any fermented food product or the humans, we need to be sure that they are fully free of the negative sides.
When you have sequenced the whole genome, do you see genes involved in competence? I mean that
yes,
genes.
Yes.
You do.
Yes, we do.
So, okay, they do. So, do you know whether it is natural competence?
Look like they're natural.
Yeah.
We was actually we published last year on these strains, and we speculate that most probably a link between Africa and Brazil. This is during the slave trade. Most probably they was transported from one to another continent, but they're very stable. Once they enter the specific environment, they really stay forever.
Okay. Thanks, Slavi.
Hi, Slavi. Can I interact without asking organizers? I don't know whether they will allow me or not. I have a different questions.
Yes.
All the probiotics and other aspects we have seen, but you know, world is moving toward the corona. So we are crazy with these viruses. I know your past experience. You have done some of the antiviral potential of these lactic acid bacteria through co-aggregation, or they aggregate with the viruses. Or do you feel some line of hope to see the activity of such probiotics or bacteriosense against viruses in general, or coronavirus particularly?
I think I will be very speculative if I say something like we can kill corona or we cannot kill, because technically I never work with corona. I work with the colleagues with another type of viruses, and based on the literature and based on the following the information that's coming almost every day, even Professor Suvorov recently, he has a very interesting interview and showing some potential of some lactic acid bacteria and fermented dairy products could be used as some of the way to treat and to prevent these infections. Well, we know that some lactic acid bacteria can trap the virus. We know this fact. This is already published. We know that some antimicrobial proteins, they can interact with the virus. We know that the protease inhibitors, they work very well, especially this is extremely high numbers of articles reporting on protease inhibitors that can really block the replication of different viruses, including even HIV virus, including Zika, polio, whatever virus you can imagine. So the people working in that, it's not that no one work. The question is how we can suggest something, but this really will be realistic to produce these protease inhibitors, and these protease inhibitors can really do their job. Because it's one thing that we can do in the laboratory, different things that we can apply it in the real system. From another side, there was several papers showing that several plant extract could be having the antiviral activity. So again, this is some area that really needs attention. And I think in the moment, we need to think about the post-COVID era, because virus, it's arrived, the virus will go, but we need to take care for these people that need to recover. And now it's coming all the information that we have for treating the patients after having influenza virus. So we can use this as an example, how we can treat them and help them to better recover, and to go back to the normal. Because I have a colleague that had a coronavirus, and he was a very young individual, a very healthy lifestyle. He almost died from corona. And he mentioned a couple of days ago that his lung system is still recovering. Will take a long time to go back to the 100%, maybe never go back to 100%. So now the question is how we can use the probiotics to accelerate the process of the recovery, not to treat the virus, but how we can use them to recover process quicker. So the people that develop the vaccine, they do their job. I'm actually looking the way to find how to can have the Sputnik 5 to vaccinate myself. This is something already done. But now we need to think how we can help the people to recover better and quicker and go back to the normal life. Because we need to remember the probiotics are not a medicine. They're a supplement. They can help the people to have the better, healthier life. So if we can have appropriate probiotics that we can provide to the consumers based on our experience with another viruses, this will be a very good idea.
Thank you, Slavi. Thank you very much for elaborative response.
Sorry,
hello, Slavi.
Hello, Mince.
Do I have time?
Well, since I'm chairing the session together with Professor Laptev, we can do what we want.
Sorry, Santosh, I hope I didn't cut you off. Are you done?
One quick question, Mince, because we need to move to the next presenter. But
okay, I can see, it's a very quick one. I can see that during your screening stage, you use RAPD and you only use one primer. And I can see that there are so many other strains that look quite unique. So how come you only pick these two?
Well, in fact, we don't use only one primer. We use a different, I think was 5 RAPD and 1 REP primers. Because the rules in this kind of work, if you have something that look equal, if they look different, they're different. But if they look equal, then we don't know. So we need to check at least 3 or 4 different primers. But I just put a picture of one of them. But in fact, we have used the several. Because we wanted first, our initial idea was to screen something as a good bacteriocin producer. And we have it. The strains was really good bacteriocin producer. But when we go to explore their safety, we found that actually very bad guys and actually dangerous.
Okay, thanks, Slavi.
It's a simple method. You can always use a simple method. If you want, you can do full genome sequence. You can compare the entire sequences. But in the beginning, we can use it as a preliminary way of the selecting.
Thank you, Slavi.
Professor Laptev, can we move to the next presenters? You agree?
Dear Svetoslav,
okay. Okay.
Please, you are also chair, and it is your time.
Okay. Actually, you can answer to me in Russian. I can understand, but still difficult to speak. I hope you understand my initial presentation in Russian. I hope. Okay, but for me, it's a pleasure to present the second presenter from this station, session, Gwenel Yan. It's a colleague that I know for a long time now. He worked on the French National Institute for Agricultural Research, INRA, in France. And he's working already several years on the Propionibacterium and showing their potential beneficial properties. Today, the presentation of Professor Gwendolen Yan will be the K role of surface layer protein, SLPB, in the probiotic properties of Propionibacterium freudenreichi. And I always asking him, can you maybe change the name to something much more easy to pronounce? So Gwen, the floor is yours. We're looking for your presentation.
Hello, everybody, and welcome to ICOBM 2021. So my name is Gwenel Jean. I'm going to show you the key role of surface layer protein, SLPB, in the probiotic properties of Propionibacterium freudenreichi. First of all, talking about the key role of the first colonizers, there are, just after birth, children are colonized by good bugs and by bad bugs, as seen on this slide. Depending on the health status, depending on disease and infection, on many different criteria, the microbiota establishment will favor either good bacteria like bifidobacteria, Faecalibacteria, for instance, and lactobacilli, and this will lead to health, or bad bacteria such as Enterococcus faecalis, for instance, and this will lead to disease status. Also, the main thing is the dramatic effect of the absence. If you are this kind of baby with which is bottle fed, in contrast with this happy child who is breastfed, you will have the absence of bifidobacteria in particular. Bifidobacteria, and especially the subspecies infantis, are champion colonizers of the infant gut. Bifidobacteria display anti-inflammatory activity, which is well characterized now. Bifidobacteria decrease the risk of necrotizing enterocolitis, and bifidobacteria increase vaccine response in the children who have bifidobacteria in their gut microbiota. Also, among the early colonizers, Propionibacteria were more recently described by the team of Mansour Mohammadzadeh. Propionibacteria are enriched in breastfed infants in comparison with formula-fed infants. Propionibacteria mitigate induced necrotizing enterocolitis, and Propionibacteria regulate Th17 cells, as shown in this paper. So the gut microbiota and the inflammation, there is a very important link between these two things. They are pro and anti-inflammatory bugs. As seen on this slide, the microbes within the gut microbiota talk to our immune system by using immunomodulins, short-chain fatty acids, and vitamins. In case of homeostasis, you have an equilibrium between symbionts and pathobionts. So, and this leads to regulation, and these pathobionts lead to inflammation. In the disease state, there is a loss of equilibrium, and inflammation is favored by too many pathobionts, and this leads to inflammatory diseases. An important factor in this respect are microbial-associated molecular patterns from the microbial parts and pattern recognition receptors from our parts. So let's talk about microbial-associated molecular patterns. They can look like this. This is the surface of a gram-positive bacterium, where you can see in blue the peptidoglycan cell wall, and in pink surface layer proteins, in white polysaccharides, here in red proteins, and in yellow lipoteichoic acids, for instance. This was also well depicted by Sarah Lebert in Nature Reviews, where you can see here fimbriae, glycoproteins, and different surface compounds like wall teichoic acid, lipoteichoic acids, which are represented here also, lipopolysaccharides, and peptidoglycan. So it's a world of macromolecules and glycobio. Now, from the host point of view, pattern recognition receptors include, for instance, on this figure, you can see here dendritic cells, which connect with, which recognize bacteria using lectin-like receptors and toll-like receptors. And in these immune cells, if you zoom on this part, you can see that toll-like receptors, node-like receptors, C-type lectins, and different receptors are involved in the recognition of this bacterium. Depending on the way dendritic cells are stimulated by probiotic or host bacteria, they will stimulate naive cells, which will differentiate into Treg cells, Th1, Th2, or Th17s. And this will lead to completely different immunomodulatory effects. So about Propionibacteria, can we use Propionibacteria against inflammation of the gut? Well, we started working on this with my friend, my colleague Benoît Foligné from Institut Pasteur Lille many years ago. Propionibacterium freudenreichi is an actinobacterium, and we found promising immunomodulatory effect of just selected strains of this bacteria in vitro and in vivo. And this graph shows you that the amount of immunomodulatory IL-10 stimulated by strains of Propionibacterium freudenreichi are very dependent. So we found very highly strain-dependent immunomodulatory properties in Propionibacteria. See, this led us to make cheeses with the selected strains of anti-inflammatory strains of propionic acid bacteria, which were given to mice. This is a happy mice with a healthy colon, where you can see in blue, in dark blue, the gut epithelium with the crypts here. And this is a sick bacterium with colitis induced by TNBS. And this is a control mice receiving PBS. And you can see a very severe destruction of the blue epithelium. And this is necrosis of the gut epithelium. These mice ate every morning a piece of sterile cheese without bacteria. And you see that there is no protection and the epithelium is still destroyed. And these mice here got cheese with propionic acid bacteria inside, and you have a very nice protection of the blue gut epithelium, as you can see here. So if we look now at the individual AMEO score, which represents the severity of the disease, it's 0 for healthy mice, and it's elevated for mice with colitis. And this score is reduced by eating this emmental cheese with the immunomodulatory strain of Propionibacteria. So there's a key role of surface protein of these bacteria in protection against colitis. You can remove using guanidine, you can remove surface proteins, and this is a bacterium before extraction and after extraction. So guanidine treated cells have lost their surface layer proteins, which are analyzed here on an SDS page gel. When you do that, you find in this extract protein of the SLP surface layer protein family, like SLP B, A, and E in particular. As you can see on this graph, the ability to induce IL-10 in human immune cells is completely lost after extraction of surface layer proteins. Now, on the contrary, this surface layer protein extract, when applied to French immune cells, will induce in a dose-dependent manner the release of IL-10. So surface proteins from Propionibacterium freudenreichi induce the production of IL-10 in PBMC cells. But which one of these S-layer protein is involved? To answer this question, we decided to compare many strains and to try to find a link between the variability of surface protein and the variability of the immunomodulatory proteins. As you can see here, we studied the strain dependency at the phenotypic level. So different strains, 25 strains, were put in contact with PBMCs from human immune cells purified from blood, and after 24 hours, we compare the amount of IL-10. And you can see that we have this strain inducing nice levels of IL-10, the strain 129, and strains that do not induce IL-10, that's the strain 121. And then in vivo here, we take mice, we inject TNBS rectally, we cause acute colitis, and then we measure the incidence of the disease, the intensity. As you can see here, a macroscopic score is very elevated for mice receiving TNBS. On the contrary, mice receiving prednisone, which is an anti-inflammatory drug, have a low score. This is a mice with colitis receiving the strain 121 that does not induce IL-10. It does not protect. But when the mice receives the 129 strain, you have a very nice protection, and this strain is responsible for protection. So there is a link between in vitro IL-10 induction and in vivo colitis protection. Now, all the strains were sequenced and the genomes were compared. So we tried to find a link between induction of IL-10 and the presence of specific genes. The same thing was done at the transcriptomic level. We studied the strain dependency at the transcriptomic level, trying to find links between IL-10 induction and the expression of specific messenger RNAs. At the proteomic level, once again, we linked induction of IL-10 with the presence of specific surface proteins, as you can see here. So at the end, there's a lot of statistics to put together: genomic data, transcriptomic data, and proteomic data, together with the phenotype, which was induction of IL-10. So this was done with a lot of statistics, and we found at the end, here in red, genes correlated with IL-10 induction. In green, proteins correlated with IL-10 induction. And in blue, messenger RNAs correlated with IL-10 induction. And you can see in the middle that there were 3 candidate genes, proteins, and messenger RNAs. And these were 3 surface layer proteins, including SLPB. So to investigate the specific effect of SLPB, we extracted surface layer proteins from Freudenreiche, and then we purified by gel filtration this protein, as you can see here. This SLPB protein was then put in contact with HT29 cells, so human intestinal cells, and we look here at expression of IL-10. So as you can see here, there's once again a nice dose response of induction of IL-10 as a response, as an answer to the presence of SLPB. So the proposed mechanisms for SLPB is that in this cheese, you have these bacteria, propionic acid bacteria. At the surface of them, you have surface layer proteins SLPB. SLPB act on immune cells, they induce IL-10, and this stops inflammation. So to confirm these results, we decided to inactivate the SLPB gene and look at the consequences of it. So to do that, we put part of the SLPB gene into a suicide vector with an antibiotic resistant genes, and then we transformed Propionibacterium freudenreichi, and we looked for integration of the suicide vector within the SLP gene, and this mutant was then sequenced to confirm inactivation of the gene. Now, as you can see on this gel, the SLPB protein here disappeared in the mutant strain line 2 compared to the wild type strain line 1. And then we used Western blotting using an SLPB specific antibody and confirmed that SLPB was present in the wild type strain line 1 and absent from the wild type strain line 2. So then the gene knockout changed the immune modulation. Once again, we put these bacteria in contact with HT29 cells and we looked at the immune response. As you can see on this graph, we're looking at the expression of IL-10. IL-10, compared to untreated cells, was very well induced by the wild-type Propionibacterium freudenreichi strain. And then, in the contrary, the mutant strain deleted in the SLP gene failed to induce IL-10 in these human intestinal epithelial cells. We look here at the messenger RNA for interferon alpha, and as you can see here, this interferon is induced by LPS from E. coli, but not by the wild-type probiotic Freudenreichi strain. In the contrary, the mutant lacking the SLP gene surprisingly induced interferon alpha in human intestinal epithelial cells. So we have a completely change in immune modulation. We've then looked at adhesion. As you can see on this slide, Propionibacteria labeled here with a green fluorescent dye were put together with these human intestinal cells, which were colored with the nucleus in blue and the cell membrane in red. This is once again in confocal microscopy, and this is in here shown in scanning electron microscopy. Now, if we look at adhesion, which was quantified using Petri dishes and CFU counting, you see that inhibition of the Propionibacteria to the human intestinal epithelial cells can be completely inhibited by adding anti-SLPB antibodies, and is also completely inhibited in the mutant lacking SLPB. Now, using flow cytometry, we were able to show that this is the fluorescence of cells alone. If you add the fluorescent wild-type strain, you have this nice induction of enhancement, I should say, of fluorescence. So the peak is moves to the right. But if you use this time mutant bacteria without SLPB, you use the ability to shift fluorescence of this HT29 cells peak. So no adhesion in the mutant. So what about in vivo consequences of SLPB inactivation? So we have worked on colitis, which is an inflammatory bowel disease, and this time we worked on mucositis. Mucositis is another inflammatory disease caused by cancer drug chemotherapies like 5-fluorouracil, 5-FU, and it causes mucositis, which is a complete inflammation of the gastrointestinal tract from the mouth to the rectum. So to do that, we take mice, we inject 5-FU, and then after 3 days, we kill the mice, and we looked at the ileum this time. As you can see here, you have on the left a healthy ileum with the crypts and the villi colored in I say purple. Mucositis causes a complete destruction of the villi that you see here, no villi at all, and there's also infiltration by leukocytes. So this is a model that is used to study the probiotic properties of Propionibacteria. Here, we have given the wild-type Propionibacterium freudenreichi to the mice before induction of mucositis by 5-FU. And as you can see here, you have control mice with a nice ileum epithelium. In the middle, you see destruction of the villi in the epithelium of the ileum in mice with 5-FU. And here, mice eating Propionibacterium freudenreichi were protected. On this histogram, you can see the score of the disease, which is 0 in healthy mice, which is elevated around 10 for mucositis mice. And you can see that the wild-type bacteria, Propionibacterium freudenreichi, protects the mice from induced mucositis. So what about the mutant? We did the same experiment with the mutant. Here, the 4 photos on the left are happy mice, healthy mice without mucositis, and here on the right, after induction with fluorouracil, the chemotherapy drug. You can see that there is a very severe destruction, once again, of the ileon epithelium in the case of mucositis, and you have here, with the wild-type Propionibacterium freudenreichi, a nice protection of the villi and of the crypts within the epithelium of these mice. On the contrary, destruction of the epithelium was very severe in the mice receiving the mutant. The histogram here on the left shows you that mucositis causes an histopathological score, which is elevated for the mice receiving 5FU, the control, the wild type protected, as you can see here, the mice from mucositis, while the delta SLPB, the SLPB mutant, failed to protect these mice from the inflammatory mucositis. Now, if we look at different parameters in these mice, we look here at the weight of mice. You can see that the weight loss was very severe in the control mucositis mice. This loss of weight was limited by the wild-type Propionibacterium freudenreichi strain, and the mutant failed to do so. Now, if we look at the permeability of the intestine, these mice were fed with a radioactive marker to monitor gut permeability. Control mice here and mucositis mice here. Permeability is very severely induced in mucositis mice and also in the control. And you can see here that the wild-type Propionibacterium freudenreichi protected mice, and you have a score very similar to healthy mice in the case of the wild-type Propionibacterium freudenreichi. The mutant strains, once again, failed to protect mice from this enhanced permeability. Now we looked here at the impact on spleen lymphocytes. All these mice were killed, the spleen was recovered, and the lymphocytes recovered from the spleen. This was subjected to different markers and to flow cytometry with fluorescence. And the wild-type Propionibacterium freudenreichi reduces, while the delta SLPB mutant increases Th17 cells in the splenocytes of these mice. So now if we look at the cytokines within the gut, you can see that the ratio between IL-10 and IL-12, is incredibly induced by the wild-type strain, which is the anti-inflammatory effect of this wild type. And this ratio is absolutely not enhanced by the mutant cells. So once again, more IL-12, less IL-10 when eating wild-type Propionibacteria and not with the mutant strain. So wild-type strain increases the IL-10 over IL-12 ratio, not the delta SLPB mutant. So if you want to read more, you can read this article by Felipe do Carmo in Oncotarget. As a conclusion of this talk, food, commensal, and probiotic bacteria have various MAMs, molecular, microbial-associated molecular patterns. Various immune responses, it may be very strain-dependent. We can make fermented foods like cheese with these strains, and these are the name of the students who participated to this work. And I want to thank you for your attention, and I'm ready for questions, and please don't ask tough questions.
Thank you very much, Gwen. I can see you just is with us.
Hello,
From France.
Yes.
And are you on the beach somewhere or at home?
Yes, I'm spending my holidays in the beautiful island of Corsica.
Oh, very nice. It's nice to combine work with the pleasure.
I'm trying.
So if we have any questions from the colleagues from all around the world, now it's a time to break the Gwen's holidays and ask him a questions. If no, I will start maybe. Gwen, I have always this question. We do all these experiments with animals, but in fact, mice, they like to eat cheese?
They love it, actually. It really depends on the recipe. The first experimental cheese was made only with propionic acid bacteria and at very high concentrated level and was very, very spicy. So we had to gavage the mice. But the second cheese that I have showed was with an anti-inflammatory strain of lactic acid bacteria and another anti-inflammatory strain of Propionibacterium froidinaceae. So we made a real emmental cheese. And as in the cartoon with Tom and Jerry, I can tell you that they love the cheese. So you have to give them the right dose because you can't stop them eating cheese.
And do you think that it's a hope to people on the chemotherapy cancer treatment? They can use these probiotics to make their life quality of life better, because there's a very hard side effects of these treatments.
Mm-hmm. I know.
And you show it. There were several other articles showing the recovery of the patients much more better with treatment with probiotics. But again, I was really interested. What's your opinion? Can we expect something in near future?
Yes.
As a real product on the market?
Well, we have to be very cautious, very prudent with that kind of result. Looking at the literature, there were a few clinical studies implementing anti-inflammatory strains of lactobacilli in the context of mucositis, and they got nice results with improvement of the quality of life and eating of the patients. So as you know, making clinical studies is a difficult question. You need a lot of money to do that. You need to work with clinicians, and I'm presently discussing with clinicians to start that kind of clinical study in French hospitals, but it costs something like 1 million euros to start. Just a detail.
Any other questions? I think Mike wants to ask something.
Yes, Slavi, with your permission. Thank you
of course.
Very much.
Hello. You're the president.
Well, you're the chair. So I have perhaps a two-component question, and it's partially philosophical, partially scientific.
I'm not surprised.
From
you.
The first part of my question. We've heard the very interesting, intriguing presentation on the role of the surface protein. And of course, probiotics, I think they're getting their well-deserved place in medical treatments. And we're not scared anymore when we see that, some of these organisms are genetically modified. In fact, Danone threw on the market its products, several of them, and they're labeled as containing genetically modified microorganisms, and they're sold over the counter. With these, a question. Perhaps you can elaborate a little bit on it. Would it be wise to consider expression of the surface protein in another microorganism to improve, to enhance activity and to create, super probiotic?
Well, I can answer at least a part of your question, which is a tricky question. Once we have proven that the SLPB protein was responsible for that, it then got relevant to use it as a purified compound in a pill, in a cream, in any way of, like a medicine. Now, the problem is that this is a protein with a lot of glycosylations, and especially with fucose. And if you don't have proper glycosylation of the protein, I do not think that it's going to bind to the lectin-like receptor of the human immune cells and epithelial cells. But to answer this question, purification of SLPD from wild-type propionic acid bacteria is very easy. You have one step of guanidine extraction and then one step of size exclusion chromatography in PBS or even in water. So I don't think I would go for expression in E. coli or in B. subtilis, for instance, because it's so easy to purify at the moment.
Thank you very much. You actually destroyed my second question, which related to other aspects. But
I'm so sorry about that.
No, actually, I appreciate you answering in a broader way, and I'm glad to hear that this protein is really easy to purify. So again, we would like to wish you luck with this study, and should the effect be confirmed, of course, as a purified substance, it may play more effective role as a medicinal application. Thank you.
Well, you're very welcome. Before I leave you, I would like to once again congratulate Min Se-young for organizing this such lovely meeting, this conference, even in these very tough conditions of the coronavirus epidemic.
Thank you very much, Gwen.
Thank you.
And,
hi, Gwen. Thank you.
It's so nice to hear some of you, but you didn't start your camera, Minse.
No, not now.
But
Enjoy your holiday, Gwen.
I feel like meeting members of my family again.
Yes, me too, me too. Now it's being held together by Mike.
Okay, thank you very much, Gwen. Thank you very much, Mike, to organize the family meeting again.
You're very welcome, Slavi.
And I would like to present the next speaker because we are a little bit running not on the schedule.
Sorry, I was long.
It's okay. It's a pleasure to hear you. The next presenter is coming from South Korea. It's Mexican by nationality. It's Jorge Enrique Vasquez Buccelli, and he's a PhD student, and he will present about fish flow, a non-PCR-based technique for detection of vancomycin-resistant enterococci. So I think it's time to see the presentation of Vasquez Buccelli, and after that we can make his life difficult with the questions.
Good morning. My name is Jorge Vasquez. I am a candidate for PhD at Hamdan University in South Korea, and I'm going to introduce you to my project that talks about the use of fluorescence in situ hybridization, together with flow cytometry for the detection of vancomycin-resistant enterococcus. Every day, our bodies are exposed to an immense amount of bacteria in our environment, whether through food, air, lesions in our skin, etc. These bacteria will be part of our microbiota and will bring various benefits to our organism. However, there are factors such as the use of antibiotics, diets, and or oxidative stress caused by some diseases that will provoke the selection of certain opportunistic population of bacteria that can cause adverse effects on our bodies. Such is the case of vancomycin-resistant enterococcus. This opportunistic bacteria has adapted and developed defense mechanisms to evade the effects of these agents on them. This represents a growing threat worldwide. The German National Nosocomial Infection Surveillance System determined that the number of cases between 2007 and 2016 increased approximately 54% just in these 10 years. Until today, 8 vancomycin-resistant genes have been identified. However, in this research we focused on the VAN-A and VAN-B genes, since these genes are found in plasmid form and there is a probability of horizontal gene transfer to another bacteria. The defense mechanism basically consists in the decrease of the affinity to the vancomycin receptors. The peptidoglycan precursor of the bacteria, D-alanine, is substituted for D-alanine D-lactate, reducing the affinity 1,000 times. It is for this reason that early detection of VRE infections is a factor in providing adequate treatment, and then we can reduce the mortality levels in affected patients. The World Health Organization estimated that in 2017 there were 49 million cases of sepsis around the world, of which 22% died for the infection. This number accounts for approximately 20% of all cause deaths globally, and from the patients who recovered from the sepsis, around 30% of them died within the period of 1 year due to the deterioration of organs as a result of the infections. This mortality is correlated with the time of diagnosis and response. The faster the response, the greater the probability of surviving a bacteremia, reaffirming the importance of a timely diagnosis. Unfortunately, the current methods can take up to 48 hours to give a diagnosis. On the other hand, we have the fact that the antibiotic resistance is a growing problem. It has been more than 30 years since the last introduction of a new class of antibiotic. So, it's necessary to seek alternative treatments for these drugs. One of these alternatives is the already known bacteriocins. These molecules with antimicrobial activity have demonstrated in different researches that they can be used as a treatment for bacterial infection. And in a context where low-resort vancomycin or other broad-spectrum antibiotics don't have the desired effect, bacteriocins in the future could help to reduce or eliminate infection in affected patients. To solve the two previous mentioned problems, we proposed fish flow as an useful tool for a quick diagnosis for this bacteria. It is based on the hybridization of a di-leveled oligonucleotide probe, which is complementary to an RNA sequence within the bacteria. This will allow the detection of the target nucleic acid sequences among thousands of individual cells in a matter of hours. It can detect more than one target gene at a time. This means that we can detect not only the bacteria, but we can also detect the resistant genes at the same time. And another advantage is that we can detect since 10 to 4 bacteria in one sample. The fish flow process is as follows. The bacteria of the sample of interest will be purified and fixed. Once this process has been carried out, the bacteria are permeabilized, which will allow the entry of the probe that has a fluorescent marker. This probe will hybridize specifically with the genetic material of the target bacteria by increasing the temperature on the sample. Subsequently, the non-specific binding will be eliminated by washing, and finally, we will be able to observe the fluorescent signal of the labeled bacteria by flow cytometry or microscopy. So, our goals for this research is the characterization of VRE from patients with bacterial infections, then the isolation of bacteriocin with anti-VRE activity, and finally standardize the fish flow for an accurate and prompt detection of VRE. To achieve these goals, we collected 40 samples from patients with bacterial infections in hospitals from South Korea. Then, we cultured in MRS plates and selected the CFUs for their morphology. Later, we characterized the bacteria by rapid PCR and 16S RNA sequencing. Then, we confirmed by PCR the presence of the BAN-A and BAN-B resistance genes. Subsequently, we analyzed their sensitivity to bacteriocins, and finally, we carried out the detection of the BAN-A gene by fish flow. As results, we have that The antibiotic susceptibility test showed that 12 CFUs selected for their morphology were resistant to vancomycin. These tests were carried out according to the EFSA regulations. The 12 previously mentioned bacteria were characterized by rapid PCR and 16S RNA partial sequencing. We were able to differentiate 6 different strains belonging to the genus Enterococcus. Then, the mix of these bacteria was evaluated against purified bacteriocins from different bacteria, where it was observed that the BREs were particularly sensitive to bacteriocins 35 and 36, isolated from Pediococcus acidilactici. Next, we want to observe the interaction of vancomycin together with the bacteriocins versus BRE. We performed a viability test with rubidium iodide in flow cytometry to observe the survival of the bacteria when mixing these two components in different dilutions. We observed that there was an inhibition in the growth of BRE in vancomycin concentration of up to 32 micrograms per milliliter. These data suggest that there is a synergistic effect when you combine these two antimicrobials. To reaffirm these results, we carried out the same experiment, but this time we used spectrometry. We obtained similar results to the previous experiment, where we observed that the optical density was higher where there was lower concentration of both antimicrobials. Once the sensitivity of BRE was known, we wanted to know if the entire population in a strain carries the BNA plasmid. So, we created mirror plates of BRE19 strain, and using a previous plate inoculated with this strain, we took 100 CFUs. Each CFU was grown under conditions with antibiotic and without antibiotic. The results demonstrate that less than 30% of the CFUs growth in a medium with antibiotic, which would indicate that the presence of the resistant genes is there. So, they were selected for the next experiment, and those that didn't grow under these conditions were selected as susceptible bacteria. Finally, fish flow was performed using the PNA-VANA probe with a non-resistant ATCC Enterococcus strain. In this first image, the control is shown where the probe was not incorporated, and therefore there was no fluorescent signal. Then, in the next picture, is the same bacteria with the probe included. However, as there is no presence of VANA, there is no fluorescent signal. Then, we carried out the experiment with the previously selected susceptible and resistant colonies originated from BRE19 strain. The absence of the plasmids was observed in the susceptible strains, where a low fluorescent signal was observed. While the flow fish performed on 5 of the previously selected resistant bacteria, the presence of the plasmid was detected in a frequency up to 50% of the population. The results obtained through fish flow have a similar trend to those observed in the mirror plates previously, where not all the population of resistant strains has the resistant gene, and in the populations where the gene was not detected was the susceptible strains. As a conclusion, we have two bacteriocins that have an inhibitory effect on BRE, in addition to a possible synergistic effect when they interact with the vancomycin. Also, the specific probe for the detection of VANA detected the presence of plasmid in resistant bacteria. However, not all the population in a resistant strain has the plasmid. This has already been demonstrated in another researches, where it has been reported that bacteria tend to lose their plasmid in non-stressful conditions. Finally, we can say that the fish flow is a good alternative for the early detection of BRE infections. However, it is necessary to carry out more tests to validate its functionality at clinical level. Well, is the end of my presentation. I want to thank you for your attention. And also I want to thank ICOBM for the opportunity to allow me to expose my project to the scientific community. Thank you.
Thank you very much for the presentation, Jorge. Now we have time for questions, but since we are running a little bit out of the time, maybe one or two quick questions from the audience. And if no one asking a question, I will ask. I think Mike wants to ask something.
Yes.
And Zoom. Mike, can you be first?
Thank you. I have a very quick question regarding the possible synergy. Have you done the tests such as to identify the fractional inhibitory concentration indices or isobolograms? You are planning to do that to be firm in understanding whether you really have synergy. Thank you.
Jorge, I think you need to unmute your microphone.
Hello, can you hear me?
Yes.
Okay. Hello. Yes, we still haven't do further test on it. We only make the viability test in order to check if was this synergistic effect. But this article is still in process, and we still need these further studies to really have a good vision of how this is working. We are believing that the bacteriocins and the vancomycins have different receptors on the bacteria, and that's why we have that effect.
And Zunk?
Thanks for a very nice talk, Jorge. I'm wondering, when you use the DNA probe, do you somehow open the cells to allow the probe to get in, or you just add without any some detergent? I mean,
yes. No. Well, we use first we use a low concentration of triton in order to make this permeabilization.
Yeah.
Yes, and then we make the fixation process with ethanol and paraformaldehyde.
So the cell is dead when you do the analysis?
Yes.
Okay.
In fact, there is another kind of protocol when where you can use the live cells, and it would be interesting to use it. But in this time, it's only we use the dead cells.
Yeah. The second, if I have another short one, Slavi,
yes.
Don't you expect to see more separated populations? I mean, the label and the non-label, it should be better separated by the flow. We did something similar. We are also trying to develop a diagnostic kit, and we managed to separate the labeled and the non-labeled much better than you do. So can you just somehow stress the two populations from each other better?
Sorry, I have a bad signal, but what I understood is if I can get better results if I get lower population on it.
Yeah, better resolution by the flow.
I think so. The problem is, as you can see in the population with the in the vancomycin resistance, the population with the gene is very low. So if we decrease the population of bacteria, there will be fewer cells with the resistance genes. That's why we use that one. That's high population.
I see.
And I can add something. Since I'm assisting to watching what Jorge doing the last 2 years, he worked with some other group of the bacteria and developing the probe for some other groups. And in some of the other groups, he has actually much more better separation of the signal and non-signal. So it very depends of the group of the bacteria. So we have some better results with the different, not vancomycin resistant, but with some other bacterial groups. Okay, but thank you very much, Zoom. Thank you very much, Mike, for the questions. Thank you very much, Jorge, for the presentation. And we need to move to the next one because, again, I say we're a little bit out of the time and people will be upset because we will putting them far away from the lunch. So the next presenter is Associate Professor Andrei Gorovtsov from Southern Federal University of Russia. And he will present the role of bacteria in the plant rhizosphere and the perspectives of PGPR application as a plant probiotics. Thank you very much to accept to present your presentation, and the floor is yours.
Dear colleagues, it's a pleasure to give a report here in this conference, and as far as I know, most of the reports were dedicated to the probiotics for mammals or for humans, but probiotics are organisms, microorganisms that are beneficial for the host macroorganism, and they can be beneficial not only for mammals but also for the plants. And what I'm going to tell is a brief review about the plant growth promoting rhizobacteria and their aspects as plant probiotics. First of all, the rhizosphere is a thin layer of soil adjacent to the root surface, and this thin layer is the interface through which the plant interacts with the soil environment, and for the plant organism it is as important as the gut surface for a mammal, because plant feeds through it and it interacts with bacteria which colonize the plant surface and the adjacent soil. This microbial community plays a crucial role for the plant, and further I'll try to show it. Historically, the rhizosphere was first defined by Lawrence Hiltner, who studied the microbial community of the rhizosphere and root exudation process and coined this term. He suggested that the majority of nutrients are given to the plant by the microbial community of this thin layer, and he was the first to recognize it as the most important part of the soil environment for the plant growth. The rhizosphere is a complex environment, and it can be divided into endorhizosphere inside the root cortex and exorhizosphere up to 5 millimeters around, and also the processes in the rhizosphere are driven both by microbial community and by host plant. The host plant secretes exopolysaccharides that are called mucigel, and also part of mucigel is also produced by the bacteria. Now, this environment allows the plant to grow through solid soil environment and also to gain more nutrients, and the soil conditions in the rhizosphere are very different from the bulk soil. The carbon compounds that are secreted into the rhizosphere by the plant are defined as rhizodeposition process. Rhizodeposition consists of root exudates, liquid compounds like sugars, organic acids, amino acids, and so on and so forth, the mentioned mucigel, and also the lost cells of the root tip and the root cortex. And it is estimated that some plant species may invest up to 30% of net photosynthesis production into the rhizodeposition. So why does the plant make such large investments? 30% is a very large proportion of what it gains. There is an answer. The main beneficiaries of this resid deposition are soil microbes. And for this income, they play several significant roles in the plant's life. First is the nutrients mobilization, because many nutrients are not bioavailable for the plant, especially such elements as phosphorus and potassium. Next is the root system expansion by the creation of mycorrhiza, which allows the plant to gain elements from a larger soil volume. Biological nitrogen fixation supplies the plant with a large part of nitrogen, which is the main nutrient element for all plants. Next is the pathogens protection, because there is a constant fight between pathogenic and beneficial organisms on the plant surface, and the beneficial microbes colonize the rhizosphere, colonize the root surface, and they don't allow the pathogenic species to invade. And finally, stimulation of growth by production of a large set of compounds, hormone-like compounds, and so on and so forth. First, I'll briefly describe the nitrogen fixation. It is divided into non-symbiotic, associative, and symbiotic nitrogen fixation with different efficiency. Non-symbiotic is conducted by free-living bacteria like Azotobacter or Clostridium species and many other species, and can contribute up to 50 kilograms of nitrogen per hectare of soil. Associative is more closely related to plant species of bacteria which dwell in the rhizosphere and colonize the surface, but they are not in a mutualistic symbiosis, and they can colonize a large set of species. And finally, the most well-known and the most well-studied mutualistic symbiosis, for example, between legumes and rhizobia, which can give as much as 300 kilograms of nitrogen per hectare. Due to this symbiosis, the crop rotation systems are created, and as you can see from the picture below, the fixed nitrogen can be accumulated in plant residues and used by the further grown crops, which cannot form such symbiosis. The most well-known symbiosis is the symbiosis of legumes with rhizobia, and rhizobia don't contain only rhizobium genus, but also many genus of beta proteobacteria like Burkhalderia and some other genera. Associative symbiosis, as an example, I have shown the genus Azospirillum, and what I wanted to show is that this genus can colonize very unrelated plants like laurus here and corn. Completely different biology, completely different lifestyle, but still the same genus of bacteria can contribute to nitrogen fixation in these plants. Another important question is the bioavailability of phosphorus. As all we know, phosphorus is a part of fertilizers that are not renewable. There are pessimistic prognoses that the amount of phosphates that are mined will be depleted from different studies, from 100 to 200 years, and then the limit of phosphorus nutrition will become a great problem for the humanity. Thus, we should find ways to increase the bioavailability of phosphorus, because for now, only from 10 to 30% of phosphorus fertilizers are used in the first year after fertilization. And there are many species of bacteria that can mobilize phosphorus, like Pseudomonas, Azotobacter, Bacillus, and many, many others. They produce organic acids, and from this picture you can see that the insoluble phosphorus salts are mobilized by production of organic acids, and a clear zone around the microorganisms that produce these acids is formed. This is the general way of screening of phosphorus mobilizing strains. Much less is known about the mobilization of potassium. Potassium is often included in insoluble soil minerals, alumosilicates like mica or feldspar, and many others, and so-called silicate bacteria can mobilize potassium by chelation, by production of specific organic acids, but this group is studied quite poorly. Next is the expansion of root system. Here you can see that the rhizosphere takes one volume of soil, but it can be enlarged with the help of mycorrhizal fungi, and the volume from which the plant takes nutrition is expanded manifold. And of course, this can increase the yields of crop plants. A very beautiful picture shows that for a pine seedling, its actual root zone is very small, while the most of the volume that is covered by the symbiotic fungi. These fungi are usually from Glomerulum mycota, and they largely expand the volume that is accessible to the plant. And you can see that they supply not only phosphorus or nitrogen, but also water, which is very important in arid regions. I'll not stop in mechanisms of arbuscular mycorrhiza formation and a picture showing the symbiosis. Another important question is the biological control of different pathogens. Here, only pathogenic fungi are presented, but nowadays a lot of beneficial microbes are used not only for biofungicides, but also for bioinsecticides like Bacillus thuringiensis, for example, and to control not only pathogenic fungi, but also to control bacterial species that are pathogenic to the plant, host plant. Many antibiotic-producing strains of bacteria are selected, namely, very often it's aerobic spore-forming bacteria from genus Bacillus, Penibacillus, and related genera. Our lab also studies the biological control by spore-forming bacteria, for example, controlling Fusarium wilt and some other diseases. And plant growth promotion. Plant growth promotion has several mechanisms. I, of course, can't stop specifically on all these mechanisms, but growth enhancement can be achieved from different sides. For example, from production of bioactive compounds like indolyl acetic acid, from lowering the level of ethylene, from improved nutrition, from reducing osmotic stress by production of different osmolytes by bacterial cells. Generally, the microbiota of roots play a crucial role in enhancing the growth of plants. Here is only one mechanism shown based on blocking the mediator of stress in plants, abiotic stress. It's the substance called aminocyclopropane carboxylic acid, which is then converted to ethylene, causing the retardation of growth and decrease in yield due to abiotic stress of various kind, like drought, salinity, or maybe pollution, and so on. And it is shown that beneficial microbes can release this stress by influencing the pathways that are present in host plant. Another way of growth stimulation. To conclude this brief review, I would like to quote a very well-known Russian scientist, a plant physiologist, Andrei Fominsin. Back in 1890, he said, "It is reasonable to pose a question. Will the successful crop cultivation in the future be reduced to the adaptation of the soil for flourishing development of microorganisms in it?" Now, in the 21st century, we return to this idea. And all over the world, the biological farming is on growth. And we try to use beneficial microbes to improve the plant growth, to improve the nutrient condition for crop plants. And we can conclude that bacteria, beneficial bacteria, plant growth promoting rhizobacteria can be for sure considered as plant probiotics. Thank you for your attention. Please.
Thank you very much for the presentation. Any questions?
Yes, I have a question. I'm Tatiana Khadikina. May I ask a question to
yes, please.
Our colleague? I'm Tatiana Khadikina. I'm associate professor of First Moscow Medical University, named after Ivan Mikhailovich Sechenov. I want to ask about human beings. If we eat these plants, what may be with human beings? Because we have some biological chains, and through these biological chains, these plants can get to our table and then to our stomach. What might be with human beings if we eat these plants, that had been influenced by probiotics and other biological and some substances?
Thank you for a question.
You understand me? I can in Russian.
Of course, I understand you. Thank you very much for your question.
По биологическим цепям до нашего стола доходят эти растения, и как могли ли вы проследить, что может быть с человеческим организмом в этом случае? Please answer.
Thank you. I'll try to answer in English.
Yeah, thank you.
In fact, at least the part dedicated to mobilization of nutrients can help to decrease the use of mineral fertilizers. As far as all we know, the overuse of mineral fertilizers can be harmful for human beings because, for example, the nitrates are accumulated in plant tissues, nitrates, they can be converted to nitrates and nitrosamines and can act as carcinogens. Another side is the biological control of pathogenic fungi, which can produce mycotoxins. And if we control by beneficial microbes, if we control these fungi, then the level of mycotoxins in crop products will decrease, which will lead to improvement of our food. Plant growth promoting bacteria are very often natural strains that are not harmful to humans, and they can only improve the crop production.
Thank you.
Thank you for a question.
Can I add something? I just want to add something. Of course, you can use these microorganisms, but of course we need to be sure that they are safe in order to don't provoke any problems. And please, Professor Laptev.
Yes, my question is, what do you think about difference of terms, plant probiotic and traditional term plant inoculant?
Thank you for a question. It's a pure question of terminology. In my view, if we use a wide term for probiotics as bacteria that are beneficial for the host organism, then we can use this term and expand it to plants also, because any multicellular organism can be involved in the symbiosis with bacteria. Then we can consider these bacteria probiotics. Of course, we can use the term PGPR or PGPB. They are older terms, but why should we exclude the plants from the range of host organisms? We can use unified terminology. Thank you.
Thank you very much. Actually, I think since we're running out of time, we need to stop the discussion at this stage. If you have more questions, you can always email to the conference website, and then they will be transmitted to the authors. And I want to ask for your understanding that we're going to move the next presenter, Tatiana Minkina, for after lunch. So don't keep you away from the lunch, because most of you like the probiotics, but the good lunch is always something better than the probiotics. So enjoy your lunch. Thank you very much for the participation of everyone on this session, for presenters, thank you for the questions. And Mike, with your permission, I want to close this session. Let's have a lunch, and we can meet again around 2 o'clock Russian time after lunch for continue with Tatiana Minkina and the rest of the presenters from the program. Thank you very much again, and see you in about 1.5 hours. Mike, any questions, comments? Okay, so see you in 1.5 hours. Thank you very much to everyone.
Dear colleagues, allow me to introduce a report of Professor Tatiana Minkina, Federal Rostov Agricultural Research Center, Russia, the microbial communities of highly polluted soils as a source of plant probiotics. Please, start please.
Good morning, dear colleagues. My presentation devoted to the microbial communities of highly polluted soil as a source of plant probiotics. The extremely polluted environments are characterized of unique microbial communities that are poorly studied. Under the extensive load of anthropogenic factor of selection and fast evaluation take place that leads the formation of bacterial strain with high level of metal resistance. This strain can be beneficial for plants and can be useful in remediation. The object of the study are the spolic technosoil and microbial communities of the dry lake Atamanska. Lake named Atamanska is located in Kamensky district of Rostov region, located in the south of Russia, where high pollution level were deposed more than 50 years ago due to the waste from the industrial enterprise located near the lake. On the slides you can see the properties of the soil with the high level of metals. Up to 80,000 ppm of zinc, more than 10,000 ppm of lead, and more than 50 ppm mercury, more than 50 ppm arsenicum, and more near 1,000 ppm copper accumulated in the lake sediments. This extreme polymetal contamination has led to the formation of spolic technosoil with unique mineralogical and microbiological properties. We use several methods to study the object. The speciation of zinc and copper in technosoil, the priority polluted, was studied by the combined analytical method based of method of sequential extraction procedure and synchrotron techniques, including X-ray absorption spectroscopy, X-ray diffraction, and scanning electron microscopy. The microbiological community or structure was studied with the use of metagenomic approach. The relative abundance of microbiological phyla was determined by NGS sequences of the 16S RNA genes. The metal tolerance bacteria were isolate of metal enriched media and rich media and further selected by gradient plate method. The biochar from sunflower husk prepared at the department of our university was use of inoculation by selected microorganism. The porous surface area of the biochar reach up more than 1,500 nanogram per grams, that make it sorbent suitable for microorganism inoculation for the remediation. A model experiment, a model experiment was laid to determine the effect of selective bacteria strains of plants growth. Spring barley, the test plant, was grown of spolic technosoil, and the bacterial strain we used for remediation together with biochar or separately. Firstly, let me introduce the results of our studies. Fractionation of zinc in the technosoil unrevealed a high degree of metal fixation with ferromanganese oxide and silicates. Copper is characterized by the accumulation of residual and oxidate fraction. The EDX spectra revealed that the mineral concentration are higher than the detection limit. The main concentration of zinc phase is represented by the sulfate goslarite produced by oxidation of some zinc minerals. It has been established that the zinc can be made both agent phase and bond with clay minerals or carbonates. And the high copper concentration was recorded only in the bonds with clay aggregates. And the study of microbiology communities of the technosoil revealed that despite of the high level of contamination, the diversity of microbial community was high. At a relatively low level of contamination, a dominant phyla were actinobacteria, protobacteria, and chloroflexi. With increase of contamination level, the proportion of firmicutes increase while the abundance of chloroflexi decrease. However, the most significant change will observe of the minor phyla. The exploratory factor analysis has shown that the most essential phyla were Gamma Timonidus, Vermicomybetia, and Nitrosprea. The firmicutes is placed on the opposite sides of the factor analysis. Of the factor analysis, this suggests that the firmicutes have a high potential for adaptation to the heavy metal stress. And the representatives of the firmicutes phylum named Bacillus genus were selected as a potential metal-tolerant strain for the use of bioremediation. The strain was selected on nutrient agar supplemented with zinc salt and future selected of gradient plates with increased zinc content on the site. The most potent strains were identified as Bacillus cereus, Bacillus palmyrus, and Bacillus atrophaeus. The mixture of three most potent strains were used in the model experience alone and together with a porous carbon-sorbent biochar. As you can see, the strain effectively colonized biochar granules, forming a biofilm inside its pores. The colonization of biochar porous with bacterial strange is presented on the slides. The biofilm of bacteria inside and porous of biochar arose among the channel in the membrane like structures around the biofilm. And the results of modern experiments have shown the application of the bacteria, and especially bacteria together with biochar, improves the growth valley plants in spolic technosoil. The height of plants, root length, and driveway will increase compared to the plants grown in polluted soil. The study of leaves' anatomical and morphological properties had shown in the leaf of plants grown of contamination soil there was a violation of the integrity of epidermal layer. The number of cells in them was reduced, while the intercellular space of parenchyma was increased. The application of bacteria reduced the observed toxic levels. And the same was seen in the ultrastructure level. The pollutant impact the plastic in the leaf parenchyma, but the use of bacteria or biochar and bacteria mitigated the stress, and the change were less pronounced. The positive impact of the leaves anatomy and ultrastructure are reflected in the plant physiologic response as well. The maximum quantum yield of photosynthetics too was sharply decreased in contaminated soil, but improved with the help of bacteria application. And thus, the observed results allow us to conclude that the application of metal-resistant strain isolated from highly contaminated soil and the inoculation of the biochar improved the plant's growth and elevated the metal-induced stress of morphological, ultrastructural, and physiological level. Thus, these bacteria can be considered as plant probiotics and can be used for remediation of polluted territories. Thank you very much for your attention.
Thank you. Thank you for a very interesting report. Have any participant any questions? Both online group, both real group.
My greetings, the members of the conference. I'm in connection. I'm very glad to see all of you. I present only the first days of our results, and we are going to prolong to study the properties of the bacteria, probiotic properties of studied bacteria.
Will you, I have a question. Will you speak about what kinds of, what phylum of bacteria do you prefer for inoculations in your experiments?
Yes, we study inoculation of biochar with this bacteria before, of course. We studied the group of bacteria, and then we inoculate of our biochar with made in our department of Rostov University from sunflower husk in 700 temperature. This biochar made.
And what about genera of bacteria?
What do you mean?
Bacillus, lactobacillus.
Bacillus.
Bacillus. Thank you.
Bacillus, of course.
Thank you. Thank you.
You're welcome.
If there are no, let's move to next reporter.
Thank you for your attention.
It is hard for me to Anuj Rajan. Will you please? Please, microphone. We don't hear you. Will you please?
Am I audible? Good afternoon. Good afternoon, respected chair, delegates, faculty members, researchers, and my dear colleagues. I'm Dr. Anuj Ranjan, postdoc fellow at Southern Federal University, Russia. I'm going to present on prospect for the use of probiotic drug to improve the condition of soil and plants. The background of this plant probiotic has been already presented by Professor Andre, and I am going to add a little more in this. So, the Global Agriculture Productivity Index, GAP, reveals that in last 5 years, there's no such accelerated growth has been found in the agriculture sector, and that could be insufficient to provide a sustainable life to 10 billion people by the year 2050. And the reason behind this is the threat to agriculture, such as degradation of soil and contamination, climate change, and deforestation, etc. The only way we see the way forward is having a sustainable agriculture growth. Plant growth promoting rhizobacteria, we also referred as plant probiotic microorganisms, have evolved the ability to improve the soil quality, protect and enhancing the agriculture produces, and they can be assessed by either culture-dependent method or culture-independent method. Culture-dependent methods are conventional methods where we use phenotypic characteristics, ribosomal DNA, PS, PCR, fingerprinting, etc., to identify the bacteria. And we have now come up with a culture-independent method that has been there by analyzing direct DNA analysis from environmental samples. We can use PCR, DGG techniques, ribosomal RNA clone libraries, and also through SD sequencing platforms like pyrosequencing, Illumina, etc. And the culture-independent approaches where the microorganisms are detected without cultivation based on extracted DNA samples. And why do we consider probiotic as a growth promoter? Probiotic microorganisms have, especially PGPR, have direct effect by nitrogen aggregation, phosphorus or iron fixation, production of plant hormone, and indirectly by screening of antibiotic, secreting the antibiotic, enzymes, and antimicrobial or pathogen immunity volatile compounds. They reduces or prevent several phytopathogenic species from having deleterious effect. There's one interesting study by Islam et al. 2016, who has presented a beautiful study on the effect of probiotic on overall growth of plant, and that has been presented in this graph showing effect on germination, vigor index, shoot length, shoot fresh weight, etc. So this represents that how PGPR can enhance overall growth of a plant. And if we talk about diversity, Zulagwa et al. 2020 have presented a great work showing the diversity where 7 microbes have, although many have overlapping, probiotic properties like production of plant hormones and iron solubilization, siderophore production, but 7 of them have been found out of 101 samples that has all 4 characteristics, and majority of them have been dominated by rhizobium, followed by Bucardia, and then Enterobacter and Pseudomonas. And if we talk about the availability of bacteria and nutrient availability or nutrient uptake, then the model presented by authors like Zager et al. 1999, Marshall et al. 2011, and Yang Crowley 2000, they have stated that there is highest chance of having abundance of bacterial abundance in elongation zone and root hair zone. However, the nutrient availability is highest in root hairs. And if we look forward towards the nutrient acquisition, the most requirement nutrients are nitrogen, phosphorus, and potassium. And these PGPR have diverse microbial ecology, and soil enriched with better organic matter requires less fertilizer because they try to mobilize those nutrients in the root system, and plants easily assimilate those nutrients. A paper has been presented by Pamela Calvo, Plant Growth Promoting Agriculture Induces Changes in Arabidopsis thaliana Gene Expression of Nitric and Ammonium Uptake Genes. So it's just not the mobilization by enzymatic catalysis or anything else, but it can also even alter the gene expression, which enhances the uptake of ammonia and nitrates. For example, if we talk about nitrates, then AT-NTR genes are modulated by PGPR, and it helps to assimilate the nitrate, and also AT-AMT2, and that helps to assimilate these ammonia by the root system. Similarly, phosphate solubilization. Phosphate solubilizing microorganism, that is PMS, they secrete organic acids like they act on inorganic phosphorus, most commonly complex with calcium, iron, or aluminum, and solubilize phosphorus uptake the plant. One of the key enzymes which play a role in the uptake and this method is phytase that releases PMS, and it also helps reactivate reactive phosphorus from organic compounds. The PMS, with the help of organic acid, also minimizes the metal toxicity among plants by turning metal atoms into immobile complexes or chelating them into mobility. And some of them have been even developed as a commercial product for biophos in Sri Lanka and agri-life in India. They are being commercialized for phosphorus solubilization bacteria. If we have a look of the phosphorus solubilization in different nutrient conditions, then maltose enriched media is allowing highest phosphate solubilization microgram per ml. Similarly, there's one more called hydrogen cyanide production. This cyanide is supposed to be toxic, and how it plays a crucial role in plant pathogen production. Like cyanide has tendency to form stable complexes with essential metal elements like copper, iron, and manganese ions. And these ions are mostly in the protein function. They play a key role in the protein function. So when these ions are being chelated with the help of hydrogen, reacting with the hydrogen cyanide, it forms a potential toxic substance. And it also binds to electron transport chain, thereby disrupting the energy supply to the cells, which leads to death of many organisms. Many bacteria that can produce Xn, including alkaligens, aeromonas, bacillus, etc., and rhizobium also. The comparison of inhibition of growth by phytopathogen fungi by HCN producing and non-HCN producing bacteria have also shown that inhibition factors exhibited by the HCN producing hydrogen cyanide producing bacteria are higher. And siderophore, one of the most important properties of PGPR bacteria, because the siderophore helps in iron uptake by the plant. And apart from phosphorus, iron, and zinc, it can be easily restricted to the crop plants. Iron and zinc can be presented in the soil, but may not accessible to the plants. They are in the labile forms. Siderophore producing microbes function as effective probiotic, and they help in chelating those atoms and make it access to the plant. If we have a look at zinc solubilization, the plant is zinc is a very much required element by plants, and these probiotic bacteria have ability to mobilize zinc also. It enables absorption of zinc, thereby increasing the yield of several crops, including rice, wheat, and soybean. And the mechanism of zinc immobilization is very similar to those like iron mobilizers or phosphate mobilizations. And there are many gram-positive and gram-negative bacteria. They help in solubilization of zinc. Effect of zinc solubilization bacteria by Pseudomonas pumilus and pseudo-alkaligens on accumulation of soluble sugar, catalase, and peroxidase on paddy under normal stress condition that was exhibited by Jha et al. 2019. And then another aspect of PGPR with respect to plant is communication between plants and probiotics. That does not nutrient ability is not done, and the PGPR also establish communication between plant and probiotics. And how do they do that? And they actually secrete some plant hormones like indole-lactic acid, cytokinins. These are plant growth promoting hormones or some similar compounds. And Pseudomonas fluorescens produces cytokinins, and that play a key role in the growth of many crops, including winter wheat, winter radish, mushroom, peas, etc. Some signaling. How does a plant establish signal with those microbes, and why these are important? Because some volatile organic compounds are being released by plants, and that is being received by those microbes, and polyamine, abscisic acids, lumicrome, riboflavin. These are the signal molecules that helps to colonize and keep the rhizosphere very healthy and enriched. Root exudates as a plant to microbe signal. It's one of the most important part of PGPR. The rhizosphere is a hot spot for soil microbes because the compounds secreted by the roots are important signal for microbes, as they can either attract or repel microbes to the plant. And it also provides a mechanism by which plant may control their root associated microbiota composition and abundance. And these exudates are mostly sugar, amino acid, organic acids, and smallest compound of complex chemical metabolites. They help to attract unique microbes to the rhizosphere, and some examples are jasmonic acid and salicylic acid that are reported to involve in the interplay between roots and microbes during the first colonization event of the bacteria. To sum up these, PGPR characteristics, you can visualize in this figure, showing nitrogen fixation ability, phosphate solubilization, 604, antimicrobial antioxidants, and ACC deaminase, and then root exudates, plant growth promoting rhizosphere microbes, and then key nutrient mobilization. All these are key properties of PGPR. And it also helps biocontrol of pathogens in both abiotic and abiotic conditions. Like if it is abiotic tolerance, then it has helped plants in the left side biocontrol efficacy of Bacillus amylolepticus against Rhizoctonia acheloni in dual culture method and broth condition. So it has shown excellent growth. The picture is showing. And if we talk about fungal pathogens, it has shown excellent growth against this fungal pathogen. And to conclude, PGPR is very essential for plant growth and development with no negative side effect. And the productive efficiency of a specialized PGPR may be further enhanced with the optimization and implementation according to the prevailing soil condition. In future, they are expected to replace the chemical fertilizer, pesticides, and artificial growth regulators, which have numerous side effects to sustainable agriculture. But it's still questionable. We'll see in the gap area. The important advances on plant PGPR cooperation will be brought in the future by combining both ecology and functional biology approach. Now, there are some gap areas which are really important to have the attention, where we have a opportunity to work and fulfill the gaps. So identification of more novel and potential PGPR, that is one of the important tasks we can plan in the future research. And development of microbial diversity map for any region, unlike similarly like we do for nutrient mapping of soil. And urgent need of application of efficient tools and techniques for enhancement of PGPR for agriculture application. And Again, formulation is important because the laboratory condition and in situ condition both are different. So, of course, suitable and cost-effective formulations are required. We have to develop those tools which can be cost-effective. And overall goal should be development of PGPR as a reality and instrumental in sustainable agriculture. So I would like to thank organizer for giving me the opportunity, and I also thank Professor Tatiana for providing me this platform. Thank you so much.
Thank you. Thank you very much, Dr. Anuj Ranjan, for excellent communication. I wish you success. If any questions,
thank you so much.
Both online, both real. If there are no questions, I won't ask you. Do you think, what is your opinion about capacity of plant probiotic to destruct mycotoxins?
Plant probiotics are having a diverse, applications, and even on funguses, they act, and they have been reported against so many pathogenic funguses. So it's up to how information is being explored regarding antifungal aspect of PGPR, and that can actually, especially with reference to crop that may, associate some mycotoxins.
Thank you. Thank you very much. I wish you success. Thank you.
Thank you so much. Thank you.
Now we shall move to next. After, please, Роберт Храмов здесь присутствует. Towards the development of exogenous photobiomodulations on human gut microbiota.
Dear colleagues, thank you very much, the organizer of conference, to possibility speech about this problem. And I will speak in Russian because I have a huge presentation and must speak faster and faster. Sorry. Итак, я расскажу о новом подходе, который пока еще не реализован. И, в общем, количественные оценки я сделал все в своем абстракте. И я сейчас расскажу общую идеологию, чтобы было понятно, что это плодотворно. И, так сказать, это будет некоторая такая большая картина, вообще, так сказать, я бы сказал, методологическая. Поэтому извините, если я буду показывать картинки, и быстро-быстро, потому что иначе я за 20 минут не уложусь. Спасибо. Это пущено весной в разливе, где мы работаем. Так. Ну, это понятно тоже, так сказать. Кто живет в ком, еще вопрос. Теперь, что такое фотобиомодуляция? Собственно, это современный термин светотерапии или, так сказать, по-простому воздействие света на, так сказать, все живое, начиная от человека и кончая растениями или бактериями, или наоборот. И разные источники, и лазеры, и лампы искусственные, и, конечно, самое важное — это солнце. И у нас проект, собственно, вот уже несколько лет идет. Это вот все источники, которые мы использовали. Вот вы видите. Так. Ага. Сейчас приспособлюсь. Значит, это лазеры, еще гелий-неоновые, это матрицы светодиодные, это спектр излучения Солнца, это спектр ксеноновой лампы, которая наиболее близкая к ним, к этому, так сказать, излучению. И проект у нас был заявлен, это впервые провозглашен от стратегии безопасного солнца к полезному. Безопасный – это блокируемый ультрафиолет, а полезный – это мы его преобразуем в красный свет, который, в общем, благотворен для живых систем. Теперь, так, что-то не двигается. Вот. Да, и концепция вот она, собственно, здесь изложена. То есть мы используем материалы, которые стоят между Солнцем и живыми системами, которые содержат люминофоры, они преобразуют ультрафиолет в красный свет, можно преобразовывать и синий свет в красный свет. В общем, основной принцип, что мы увеличиваем долю красного света, падающие на растения. Вот видите, вот последний материал, который мы сейчас выполняем грант междисциплинарной РФФИ, это текстиль, агротекстиль, который можно закрывать поля. Текстиль можно делать и для человека. Значит, это вот обычный текстиль под ультрафиолетом, ничего здесь особенного нет, никак они не взаимодействуют. А вот текстиль с люминофорами, он уже дает вот такое свечение, понятно, да, всем. И вот мы увеличиваем долю красного света. И, собственно, что делает красный свет? Вот даже если взять, так сказать, животную клетку, то активирует около 111 генов. Понимаете, на микробных я не привожу, но я думаю, что не меньше, может даже больше. Поэтому все, что эффекты можно получать самые разные, и нет ничего ни вредного, ни полезного, есть такой принцип. Да? Главное знать дозу, параметры излучения, время, место и так далее. Но возможности для света фантастические. И вот, скажем, один из классиков нашей фотобиологии, Тина Кару, она прямо заявляла, вот у нее обзорная работа, что вместо лекарств можно использовать, так сказать, свет. Так, вот примерно схема, как растения растут. Это вот, скажем, укрывные две теплицы. Одна из них с обычным материалом, другая с люминесцентным, который сверху. И вот видите, как вообще свет распространяется. То есть даже геометрия уже другая, потому что рассеянный свет, он и туда, и сюда, и подлазит, и прочее. И результат такой, что можно вот как бы рядовые, значит, можно увеличить урожайность или ростовые характеристики. Ну вот в среднем вот такая цифра. Есть рекорды, и в 2 раза можно увеличить. Вот. Ну вот здесь вот тоже пример наглядный такой. Вот видите, вот это вот, видите, вот здесь вот плохо видно, но здесь соединены два вот этих вот образца текстиля. Один люминесцентный, другой обычный. И вот на одной грядке, видите, и температура одинаковая, и влажность, и земля, и все. Вот перец такой, а здесь вот видите, он такой хиловатый. Вот что делает, так сказать, такая добавка красного света. Ну какое же было наше удивление, когда мы проанализировали спектр, скажем, который вот солнечное излучение и то, что проходит у нас через этот текстиль. И оказалось, что мы даже не видим разницы. Вот здесь вот в красном свете, видите, в спектре должен появляться пятачок этого красного света, а мы его не видим. Но когда смотрим на лампе, то все видим. Просто я говорю, что он очень маленький. То есть смысл какой? Что даже небольшое изменение в спектре влияет так на растения. Значит, ну, чудес нет, конечно, когда мы концентрацию люминофора увеличим, вот тот же текстиль, он, скажем, с высокой концентрацией люминофора примерно в 1000 раз, вот чем я вам показывал, точнее вот 1600, то вот видите, уже свет проходит гораздо меньше, вот видите, исходно-солнечный, и появляется вот здесь вот этот пик уже. Мы его видим. То есть все, так сказать, реально. Теперь мы переходим плавно. Так. Что-то не переходит. Извините, да, все. Да. Теперь, значит, это с растениями. И, в общем, до сих пор не решена проблема. За счет чего? То ли за счет фитохрома листьев, то ли еще чего-то. И вот появилась работа совершенно из другой области, нефтезагрязнения анализировали и накрывали вот этой пленочкой, которая содержала люминофоры и которая не содержала люминофоры. И, собственно, сразу перейду к картинке. Вот смотрите, как ведут себя эти бактерии почвенные в нефтезагрязнении. Вот это вот обычная пленка, вот количество, так сказать, микробов. А это вот два типа пленок, ну, два разных люминофора, и вот ведет себя таким образом. То есть тут 2-3 порядка выше. Понимаете, вот это тоже загадка, но это вот как бы факт такой уже касающийся микробиологии, и, так сказать, надо с ним разбираться. Пока не ясно, но вот это факт, как бы диссертация защищена, это хороший институт, институт нефти, Томский, так сказать, университет, доверять можно. Да, ну почему вот это вот такое вот волнообразное движение? Потому что на самом деле довольно сложные там идет борьба, так сказать, не на жизнь, а на смерть. Вот, скажем, бифидобактерии ингибируют рост Е. коли, а Е. коли, наоборот, стимулирует рост бифидобактерий. В общем, такая ситуация получается в биофизике волки и зайцы, когда, так сказать, волки съедают зайцев и сами, так сказать, начинают вымирать. И потом зайцы размножаются, их опять съедают волки. В общем, примерно вот такая. Это объяснение, почему колебательный. Так, теперь, значит, переходим. У нас была работа из другого плана. Мы тестировали состояние физической работоспособности, то есть общая характеристика организма, по плавательному тесту. И что мы увидели? Да, и тестировали, опять же, вот полезное солнце. Вот, собственно, плавательный тест. Мышка плавает с грузиком, как только начинает тонуть, ее вынимают. А вот мы ее облучали искусственным солнцем, тут киловатт, через вот материалы, которые люминесцируют. И что мы видим на мышках, в отличие от растений? Вот видите две линии. Это две линии мышей. Одна облученная с преобразованным светом, это экспериментальная красная, а другая обычным светом. И вот сеанс облучения. А вот как они плавают до этого. И видите, через примерно неделю, тоже характерное время, да, вот запомним его, начинается вот постепенно расходиться эти две кривые, и процентов на 30-40 плавают дольше. Теперь дальше поедем. Уже некогда останавливаться. Ну, мы проанализировали, эта работа была в 2015 году сделана. Мы проанализировали сердце, митохондрии, миофибрилы, ретикулум. В основном на сердце ориентировались, потому что плавание – это сердце. Значит, бактериями тогда не занимались. И одна из загадок, что, возможно, там бактерии виноваты. Так сказать, прежде всего, а уже, так сказать, физическая работоспособность – это следствие стимуляции бактерий. Теперь, вот сегодня выступал докладчик у нас замечательный с Тайваня. Так вот, эта лаборатория как раз протестировала, чем это хорошо, они разные компоненты пищевые протестировали. И вот особенно вот этот вот лактобациллус плантариум из квашеной капусты, они производят этот сапплемент. То есть из одной лаборатории мы получаем как бы относительно друг от друга. Вот посмотрите теперь по эффективности. Вот это контроль. 100% мыши плавают предельно. Это наш свет делает 30-40. А вот смотрите, что делает лактобациллус плантариум. Это в 5 раз больше. Значит, я, когда эту работу увидел, то я просто в ужас пришел и понял, что это подарок, так сказать, надо им заниматься. И следующая работа у них была, вот оказывается, капсаицин из жгучего перца то же самое дает. Ну, он сожжет слизистую, если у кого-то, так сказать, слабые. А лактобациллус плантариум, он никак, так сказать, не повлияет на наше здоровье. Вот, поэтому бактерии могущественны и в плане общего состояния. И вот подход, который был, да, скажем, на сегодня, чтобы стимулировать эти бактерии светом, то есть вот предшественник этой работы, это вот такая таблетка, которую заглатывать надо, и она, так сказать, включается во влажной среде и стимулирует бактерии, как утверждают авторы, и они недалеки от истины, потому что, скажем, вот тоже диссертация была защищена, показали, что контрольная и опытная группа, одни глотали, другие лечились обычными тех, кто лечили и пробиотиками, и прочими, у них дисбиоз практически, так сказать, слабо поддавался лечению, а те, кто глотали вот эту таблеточку, у них, в общем, результаты были... Да, зажигалась она в диапазоне где-то красного, вот так вот цвета тоже, 500-700 нанометров, красно-оранжевый. А те, кто глотали эту таблетку, значит, у них, вот смотрите, как меняется состав Е. коли и лактобациллы. Видите, тут лактобациллы меняются там в миллион раз. Потому что, когда дисбиоз, то там вообще доли процента от нормы остаются. Поэтому вот подход хороший, но глотать надо, потом ловить таблетку или выкидывать, так сказать, это довольно дорогое занятие. И, значит, мы пошли дальше. Вот смотрите, как Е. коли реагирует на свет, скажем, это скорость роста от дозы света. Вот смотрите, это вообще тоже, так сказать, сказка. Вот смотрите, вот этот вот пик максимум при малых дозах, это примерно доли секунды, если взять интенсивный солнечный свет, то это доли секунды. Ну, естественно, там у нас никакого солнечного света нет, он гораздо меньше по интенсивности. А следующий пик, вот здесь вообще нет ответа. А следующий пик, он только вот здесь вот. Это когда вот, так сказать, вот длинный волн здесь указан, здесь и красный свет, и инфракрасный. То есть вот это вот вдохновляет то, что можно на малых дозах это воздействовать и стимулировать. Теперь следующее, еще более эффективно, если мы возьмем импульсное излучение, скажем, ближней инфракрасной области. То есть здесь доза эта, вот там была доза примерно около 50 джоулей на метр квадратный, а здесь доза 0,5 джоуля на метр квадратный. Это в 100 раз меньше. То есть понимаете, какая перспектива? Что вот даже не красный, а инфракрасный. Но толком это все, так сказать, не исследовано. Это только предпосылки. И теперь, значит, дальше. Значит, а реальна ли стимуляция микробиоты? Значит, вот смотрите, если мы будем работать в окне прозрачности для биологических тканей, а он где-то вот здесь, вот, от 650 до там 1300, вот это вот глубина проникновения, видите, вот пик вот где-то вот здесь, вот около 1000, да? Вот, то все это реально. Я эти расчеты опубликовал, можно посмотреть. А вот это эксперимент, который я проделал над собой, мне делали колоноскопию, очень приятная процедура вот с таким зондом, который светит, подсвечивает. Я очень радовался. Вот. Я радовался, конечно, не самой процедуре, а то, что у меня толстый кишечник очень хороший оказался. И пытался зарегистрировать. Вот видите, вот это вот излучатель, это 100-ваттный по мощности излучатель красного света. И пытались там, так сказать, увидеть этим зондом. Но поскольку там своя лампочка, и вот они две, то есть там засветка такая сильная, что вот это уже нельзя было зарегистрировать. Поэтому, но мы сделали другую, обратную задачу. Значит, врач мне поставил этот зонд вот перпендикулярно стенке, так сказать, кишечника и живота. И мы увидели красное пятно на животе. То есть вот это все замерили. Вот это, видите, у меня тут люксметр есть чувствительный. Значит, примерно дозы, то есть интенсивности света, они вот, я вам показывал вот этот маленький пик, да, то есть мы попадаем. То есть вот, скажем, 2 сантиметра живота можно просветить. Поэтому и количественные оценки предварительные, и эксперимент показал, что все это реально. Вот. И последние работы, которые сейчас у нас идут, мы стали работать на отдельных штаммах, потом перешли на экосистему микробиоты человека. И вот, скажем, первые результаты, еще не опубликованы, вот вы первые их слышите, значит, они, так сказать, вот сделаны, мы тест такой взяли, устойчивость микробиоты и штаммов при заморозке. То есть там примерно 50% погибает в контроле. Вот заморозишь в азоте, потом вынимаешь, а там 50% живых. Ну, мы с помощью красителей, значит, ну вот понятно, что с помощью красителей можем анализировать жив-мертв. Значит, это вот, так сказать, кривые всякие тут, такие хитрые. Это так сказать, азотный контейнер, где все это погружалось. И, собственно, значит, вот, скажем, смотрите, что на бифидобактериях Бреви мы получили. Значит, вот выживаемость, если в стандартных условиях это 50%, то мы получаем достоверное увеличение или уменьшение, когда, я извиняюсь, тут не видно время, но вот здесь большая доза уже облучения. Вот, это, значит, это было, значит, нет, здесь небольшая доза, здесь всего 11 люкс, но это все в ватт на метр мы пересчитываем. Значит, вот побольше доза, вернее, поток световой, тоже получаем точки, пока не можем объяснить. Достоверное отличие, да, и вот, а это уже, как говорится, против большой дозы, против лома нет приема, в общем, вот это очень большая доза, то есть, да, большая доза, и мы получаем угнетение, то есть у нас там меньше, чем выживает в контроле, который необлучаемый. Значит, ну вот еще, так сказать, тоже подобная кривая, посмотрели, как это все выглядит в натрии хлора, получаем достоверные изменения. В общем, вот этот вот рекорд 40%. У нас на 40%, здесь порядка 78%, а в контроле у нас 50 выживают. 40% это уже прилично. То есть смысл этого всего, что светом можно улучшить, не знаем пока как, не знаем какой механизм, но предварительное воздействие протекторное, так сказать, протекторные свойства света для консервации, может еще куда-то это пойдет. Значит, это вот уже мы сравнили, собственно, как действует 660 нанометров и 940 нанометров. Видим тоже, что есть зоны, где у нас нет эффекта, есть где есть эффект. Пока с этим разбираемся, пока, в общем, это нарабатываем эмпирику, проверяем воспроизводимость. Это в другой, скажем, фетальной сыворотке, то же самое культивация. Да, это все достоверно. Работаем в анаэробных условиях. Да, все работы до этого были сделаны, вот даже на Е-коле, они сделаны в аэробных условиях. Теперь у нас есть анаэробостат 96-луночный, так сказать, это отслеживается каждый, так сказать, там полчаса, час, мы регистрируем кривые роста. Короче, вот мы вышли на вот эту работу и, в общем, видим, что есть эффект. Пока понятия нет, за счет чего. Ну, это подобное. Подобное, вот это уже со штаммом бифидобактериум бреви. То же самое. Есть, где есть эффект достоверный. Ну, а это кривые роста мы снимаем, так сказать, как это все растет. Плотность, оптическая плотность, которая у нас есть пересчет на количество клеток. Значит, вот я так покажу, что, в общем, мы анализируем кривые роста. Видно, что свет действует по сравнению с контролем. Не очень удачный рисунок, извините. Так, это у нас, так сказать, скорости роста. Видно, что тоже есть эффекты. Разбираемся. Вот. В общем, не буду дальше злоупотреблять вашим временем. Все это работа идущая. Это вот скорости все. И, в общем, заключение следующее, что микроорганизмы очень чувствительны к оптическому излучению. Значит, и перспектива использования для медицины, на наш взгляд, очень высокая. И развитие этого метода вместо вот этой пилюли световой, оно все это реально, да. Нужно вот этот вот НИР проделать, прибор мы уже, так сказать, сконструировали. Ясно, что нужно определять параметры, здесь поле параметров, так сказать, это длина волны, доза. Потом очень оказалась важна импульсная модуляция, понимаете? То есть вот, ну, как и мы все, вот, скажем, нам, если резкая вспышка в глаза, допустим, я пример привожу, да, она значимее, чем, скажем, просто вот непрерывный свет. То же самое и для бактерий. Не знаю, так сказать, мы не знаем пока антенну, первичный акцептор, но такое есть. И, в общем, ну, это всем понятно, да, так сказать, это благостный лозунг, чем мы занимаемся, чтобы оправдывать наши деньги, которые нам дает государство, Правда, китайцы на следующую пятилетку увеличили финансирование НИОКРов в 17 раз. Не на 17%, а в 17%, да? Вот задумайтесь, да, где мы будем и где они. Ну, мы уже и так там, но еще будем глубже, как бы. Вот. И благодарю, так сказать, вот участников этой работы. Ну, тут не все, но основные, так сказать, и обсуждения с Михаилом Чикинцем очень плодотворные были, и он оценил эту работу. Благодарю за внимание. Спасибо.
Thank you very much for very interesting communication. Я хочу по-русски сказать, потому что мне кажется, что то, что вы рассказываете, это настолько интересно, и можно, если стимулировать не только бактерии, нет, ну, во-первых, бактерии можно стимулировать в ферментере, при выращивании в ферментере, или до ферментера, ну, скажем, промышленное.
Это вы предвосхищаете.
Да, да. Во-вторых, можно, нет, ну что-то мне захотелось, может быть, как-нибудь в инкубаторе яйца осветить вашим светом. Вот. Или, скажем, если вот при криоконсервации, то почему бы, вот, скажем, криоконсервация спермы? Или, скажем, каких-то, причем вот и есть проблема вот тех таких животных, достаточно экзотичных. Вот. Может быть, это резко повысит какую-то эффективность. Спасибо большое.
Спасибо.
Какие-то вопросы будут? Is there any questions from online group?
Если красный свет так стимулирует бактерии, должен же какой-то рецептор быть, он должен поглощаться. А вы просто спектр поглощения бактерий не пробовали снять?
Вы знаете, спектр поглощения, вот я упоминал кару, у них сняты, и ответственные там пики, это, так сказать, цитохром-циоксидаза. То есть один из акцепторов, я просто, это вне рамок, это мы далеко уйдем, но точно это все, так сказать, еще нужно делать. Там есть, как правило, это железосодержащие, так сказать, белки, ферменты и так далее. Вот, скорее всего. То есть, итого, есть акцептор, но, возможно, еще всякие варианты. Но я эту сторону не затрагивал, это отдельный нужно доклад делать и со спектрами, и деталями, и биохимию. Да, спасибо большое.
Спасибо большое. Thank you very much. Спасибо. Dear colleagues, let's allow me introduce Luis Nero, professor of Federal University of Viçosa, Brazil. Weissella, an emerging beneficial bacterium with promising health benefits. Will you please?
Hello, my name is Luis Augusto Nero. I'm a professor in Universidade Federal de Viçosa, is a Brazilian university. And first of all, I would like to say thank you for the organization committee for inviting me to present this lecture about Weissella. So the title of my seminar, my presentation would be Weissella, an emerging bacterium with promising health benefits. Just to start this slide, I'm presenting a general overview of the topics that we will comment today. First, I will present the general characteristics of this genus. Then we will move forward to the beneficial aspects that this bacteria present related to health features, technological features, and safety, how they present these characteristics and how the industry can use these characteristics as a potential use in the food industry. After that, I will present some results of a study that we recently published related this genus, in which we obtain Weissella strains from different food producing environments, focus on dairy specifically, and then we will comment about the biodiversity, the inhibitory activity, and also the technological properties that we found in these strains. And at the end, I will talk a little bit about the perspectives as a concluding remarks of this lecture. As I told you, most of the information that we'll present in this lecture is about to be published in Probiotics and Antimicrobial Proteins. Is a review about Weissella that we recently approved in this journal. And I think by this year, it was accepted in January of this year, and I think in the second semester of this year it will be published. And the other, and most of the information that we'll present is summarized in this mini review that we publish. Everyone, if you want to take a look at this review, you'll be welcome. And after that, I can send you, I will put my email in the end of the presentation. You can contact me and I can send you the link to obtain this manuscript. And the second one is about this Brazilian study that I told you. It was published in LLT, also in the beginning of this year. And both studies are part of the PhD work from the first author, Camila Gonçalves Teixeira, she's a student from our group in food science technology here at Universidade Federal de Viçosa. Well, this is the first, let's see, the family tree of the Weissella. Weissella is, as everyone knows, a lactobacteria bacteria, but is a relatively new characterized genus in this group. The species of this genus are characterized by short rods or coccoid form. Most of them, depending on the cultural conditions, they can present a pleoformic shape. And Weissella also is closely related. The species of this genus needs to a proper identification to identify mainly among Leuconostoc and Eunococcus. They share very common characteristics, biochemical characteristics. So a proper identification needs to be conducted to allow their identification. Nowadays, we have 25 known species of Weissella, and this was possible basically based on sequencing of 16S and 23S RNA. This slide, it was a review that Fusco et al. Published in 2015. Also, I get some information from this review. It's a very nice review that they present the basic and technological information in Weissella. And in the review that we are about to publish, we updated this information mainly in the last 5 years. So the idea was to cover this the lack of complete information about this genus from this review from Fusco et al. From 2015 and now. As we can see here, this is two similarity trees, neighbor joint phylogenetic tree of Weissella species, but based on sequencing of 16S and this gene VS gene that's important to identification of Weissella. What we can see from here is that genetically, these species of this genus are very close related. So we need to go deep in and conduct proper analysis to allow proper identification of these species. And this is a very interesting characteristic of this species, of this genus, because they share a lot of common characteristics. For the food industry, and considering the biotechnological potential of Weissella strains, we can divide these aspects in mainly 3 topics. First, about biosafety, because we know that many Weissella strains are capable of producing antimicrobial substances, mainly bacteriocides, that is one topic of our group, but also organic acids and other antimicrobial substances. The technological potential of Weissella is basically, is mainly due to the ability of their producing some substance that can improve texture on foods. These substances are the exopolysaccharides. So Weissella is very well known by being able to produce these substances. Also, they are known to produce some aromatic substances that have importance in some artisanal foods or any other fermented foods. They have many substances that they produce with these characteristics diacetyl. And in addition, Weissella were characterized as being naturally present in a variety of artisanal foods. So they have a contribution role in the development of or some organoleptical characteristics of these foods. We will talk about that a little bit more in the following, but just to understand how Weissella can be relevant with a biotechnological aspect. And finally, Weissella has also a health importance because they can be characterized as probiotics. They have some probiotic characteristics that can be used by the medical industry and also by the food industry. So this biotechnological relevance of Weissella is being more and more studied to identify isolates, to identify strains to be used as one of these applications in the food or the health industry. So this biotechnological relevance is pretty clear about Weissella. If we move forward, we can start to talk a little bit about the beneficial aspects of Weissella. If we have the last slide and get the last thing that I told you, the last slide about the health relevance, Weissella, we always have to think as Weissella as a probiotic agent. So up to date, we have 2 studies or 2 Weissella isolates, both Weissella ciparia, that they were studied aiming the direct application on human health. So these 2 studies are related to that. The first one is related to cytokine activity and to activity of immunoglobulin from natural killer cells. So Weissella ciparia JW15 were characterized as being able to improve these activities. That's important for the host defense, for the human defense. And the studies on Weissella ciparia CMU is more related to odontology. This strain was characterized as possessing ability to adhere and to colonize the oral cavity, so the mouth. And they were characterized as being able to have inhibitory activity against some important pathogens on oral pathogens as the responsible to cavities and to periodontitis and everything. So both strains were characterized and being able to improve the human health. So these 2 studies, these 2 strains, they are up to date, they directly linked to human health and can be used as a probiotics. But we have another studies that characterize other strains with also a good potential to be employed to human health. So we have these studies, several studies that characterize the antimicrobial effects of Weissella. Most of these, some of these studies are due to the production of bacteriocins. And also we have other beneficial characteristics that was characterized in Weissella strains. Related to inhibitory activity to other pathogens, for sure we have the bacteriocins ability, the ability to produce bacteriocins, but also they can improve the activity of other antimicrobial substances, for instance, antibiotics. There's some studies that describe the ability of the Weissella strains to improve the membrane permeability of the cells, of the target cells, and with this, they are able to absorb, let's say, more antibiotics and in this way, in this sense, cause the antibiotic effect. So we can reduce the levels of antibiotics associated with these acellular strains to improve the activity of these antibiotics. So this is a very important feature that is demonstrated by Ricella. Regarding other beneficial aspects, Ricella was characterized as being able to survive in the presence of daily acids, so they can survive through gastrointestinal tract to reach lower levels of the intestine. They were characterized, some strains were characterized as being able to adhere to these cells and to produce biofilms, so they can stay in this environment. And also they were characterized as being able to reduce the cholesterol levels of the host and also to produce vitamins. So all these features altogether, they demonstrate how Ricella can be an interesting probiotic candidate to be used by the food industry and the medical industry. As I said, Ricella also has a technological potential. As we said, Ricella is naturally present in many artisanal foods that we can identify all over the world. In the review by Fusco et al., they present a very nice table with a large number of foods and studies in which Ricella were identified as a natural presence in these foods. So Ricella is clearly related, has a clear contribution role in the characteristics of these foods. So they are important to determine some characteristics of these foods. And for sure is not the only one substance that is responsible for these characteristics, but the production of exopolysaccharides as being the main technological feature of Ricella is very important in these foods and to determine some characteristics. As I told you, diacetyl also is an aromatic compound that Ricella is known to be able to produce, but exopolysaccharides are being more and more studied in Ricella strains. As you can see, the main species that are being characterized are being able to produce exopolysaccharides are Ricella cibata and Ricella confusa. And the properties of the exopolysaccharides produced by Ricella are diverse as well. They are be able to promote oil absorption and emulsifying properties and improve the texture of the foods. So in this sense, the texture of the food is very influenced by the production of these exopolysaccharides from Ricella. These exopolysaccharides also has some bioprotection features because and beneficial features, in fact. They can be used as to improve the health or to protect the host. Even the food, they were described as possessing antifungal activity, anti-inflammatory, immunomodulatory, and antioxidant properties. So they have beneficial aspects in the food. And also they can be, because of that, they can be used as prebiotic substance also. The main probiotic substance produced by Ricella dextran that is very well characterized in a number of studies, as we can see in this table. Despite all these facts, these beneficial features, we have also to pay attention in the other side of the lactase bacteria. I think Dr. Todorov presented in this conference a seminar about that, about this paradoxical role of some lactase bacteria, because at the same time that they have a lot of beneficial aspects, beneficial features that can be used by the food industry or even the medical industry, they can have a dark side. They can have some virulent genes. They can induce some disease in the host. And this is, we have to find the proper balance in that. So we published some years ago a paper about that on Enterococcus. That for sure we're not talking about Enterococci here, but is a similar way to study these starter cultures. At the same time that we characterize their beneficial aspects, we have to check if there are potential foodborne pathogens, or they can represent hazard for the consumers. So regarding Weissella, this is also a very interesting collection of information, because in general, Weissella was characterized as a pathogenic agent, but only in fish, rainbow trout specifically, as being able to cause septicemia. But in general, antibiotic resistance Weissella has, the species from Weissella genus, they present an intrinsic resistance to vancomycin and kanamycin. So this is a common feature that we can find. But even presenting resistance to these antibiotics, what we usually find in Weissella strains is generally regarded as safe status. Usually they are grass, let's say, because in general, they have low frequency of antimicrobial resistance related genes. It's not so common to find a number or a high number of antibiotic resistant genes. Even when they present these genes, they don't have, the isolates, they don't have too much mechanism to transfer these genes. So they keep on them. This what is quite safe and interesting in terms of population communication that they might have in the intestinal gut or as a probiotic agent, let's say. In addition, Weissella usually don't have, Weissella species usually don't have many virulence related genes like hemolysis and other virulence genes that are important to cause disease in humans. And in the same time, even if they present some of this virulence factors, virulence genes, sorry, they don't express them a lot. And as they don't have too much mechanisms to transfer, usually these genes keep on them, so they cannot transfer them easily to other strains. So in this sense, in general, Weissella are considered as safe to be used as probiotic. For sure, what I'm saying is that they are totally safe. We need to check these features before a proposal of using or even to propose them as a probiotics or a starter culture. But in general, they don't have too much of these mechanisms. So this keeps them as usually safe to be used. And this study that we did and we published kind of confirmed that. The main focus was to check the diversity and the technological potential of some Weissella obtained from different parts of Brazil. And we get samples, in fact, 57 isolates from these three species, Weissella cibaria, Parvus enteroides, and Weissella confusa. And we get them from artisanal cheese production chains, in fact. From different parts of Brazil. We get some from the north of Brazil in Marajó Island and Pará State, where they produce some artisanal cheeses in here. They're very popular in the region. We have some isolates from Bahia, that we have this requeijão. It's a kind of cooked cheese that we have in Brazil, and also from Minas Gerais state. And we didn't get samples or isolates only from the cheese, but also from the producing environment to check the diversity in the environment and also in the end product. We conducted PFG and RAT PCR to check the biodiversity of these strains. We check then regarding antagonism to check if they're able to produce inhibitory substances against selected foodborne pathogens, not a large number, but only to check if they're able to produce some antimicrobial substance, and focusing mainly on bacteriocins. It's part of our research group. And also we check some technological features, mainly the production of acetyl, the ability to coagulate milk, as we are talking about cheese. We check the proteolytic activity, the acidifying activity, and also the production of exopolysaccharides. Among these 57 isolates, what we can see is based on pulse field gel electrophoresis and also RAP PCR, we were able to identify that even they sharing a very close relation, they have different profiles. So they are diverse. Even being present in this environment, we can see in some species the same genetic profile from different parts, indicating a kind of cross-contamination or origin of the contamination in the processing chain. But in general, we have quite diverse profiles. I cannot say genetic profile because we are talking about digestion and RAT PCR just some parts, but we have this diversity. This is one part of the study that we are trying to improve. We selected some of these isolates and we subjected them to whole genome sequencing that we are studying that in nowadays. So we'll have more results in the future. But in general, they are quite diverse. So this is interesting because I have a different strands and we have different isolates with different profiles that we can study it and to select potential candidates as a starter or even as a probiotics agents. Despite being diverse, we were not able to identify, any strain being able to produce bacteriocins. They were not characterized as bacteriocinogenic, but they were able to inhibit foodborne pathogens, most probably due to organic acids or hydrogen peroxide. Regarding the beneficial features, we selected some of these isolates to conduct that based on genetic profiles, and we were able to identify the proteolytic activity is important as we talk about cheese production. We identify some isolates being able to produce diacetyl. Even being relevant, the exopolysaccharides in these strains were not characterized, that it was a quite interesting feature. Maybe we're not able to identify this exopolysaccharide production in enough amount to be detected to this phenotypic assays that we use it, but it's a result that we have. What was more important for our result was the intense production of diacetyl, that's very important aromatics compound, and also the acidifying activity that we identify some variation. What we can highlight is the isolate 16 that was able to produce the acetyl in large amounts. These isolates were able to produce an uniform clot when it was subjected to coagulation analysis and also proteolysis, and a high acidifying activity when compared to other strains, to other isolates. So if we have to select one candidate to go deep in our studies to check their ability as a starter culture, this one is it was selected in fact, and we are going deep in the studies with it. Just to finish, I'd just like to send a message that because of all these beneficial characteristics, we are being increasingly studied, because of mainly of its high beneficial potential and low pathogenic potential. These two situations are associated, and they are interesting. I didn't present that in our study, but also we checked some virulence features, and we did find it, at least the ones that we checked that were more related to antibiotic resistance and some conventional virulence aspects. Our Brazilian study, despite of that, this technological potential, we identify high biodiversity of the isolates, allowing us to conduct further studies to improve the knowledge on waste salmon. So after that, we'd like to say thank you again for the organization comment. Thank you for hearing the data that I'm presenting, and this is my email, and please feel free to contact me to regarding anything that you might be interested. Thank you.
Thank you, dear Professor Nero. Very interesting communication. It's really beneficial bacteria. I have one question. And what kind of exopolysaccharide it synthesizes? Sorry, I forget that it was in YouTube. Thank you. Let's moving to next communication. Galina Pronina, professor, Moscow Timiryazov Agricultural Academy, improving the immune resistance of hydrobiomes in aquaculture by probiotics. Please.
Dear colleagues, the topic of our communication, excuse me, demonstration. The topic of our communication is improving the immune resistance of hydrobiomes in aquaculture by probiotics. Authors is Pronina, Shushanova, and Isaev.
Professor Pronina, will you make, please, all экран, презентацию на весь экран можно сделать?
Yes.
Thank you.
All right. У меня вот здесь почему-то плохо получается, потому что когда не на весь экран, вот. Diseases of aquaculture facilities cause significant damage to fish farms. It is important to maintain the health of fish and crayfish and increase the immune resistance of cultivated objects. One of the ways to increase immune resistance to use of immunomodulators. It is natural of synthetic substances to can have regulatory effect of the immune system. Probiotics are immunomodulators based on live microbial cultures used to correct microbial zoonosis in the treatment and prevention of a wide range of diseases. Probiotics can displace pathogenic and conditionally pathogenic microorganisms from the niches of their habitat in the macroorganism. Produce a complex of biologically active substances that can neutralize bacterial toxins, dangerous metabolites, enhance the mechanisms of non-specific immunity by increasing the antibacterial activity of lymphocytes, increasing the number of macrophages and production of antibodies. Here are following results of studying the effect of the feed additive subtilis S containing probiotics Bacillus subtilis and Bacillus licheniformis on the physiological and immunological state of carp, Cyprinus caprio L, and crayfish, Pontostachus leptodactylus. The subjects of research is 2 years old carp, 14 individuals in total divided by 7 individuals in both the experimental and control groups. And crayfish, Pontostachus leptodactylus, 26 individuals in total divided by 13 individuals in both the experimental and control groups. The fish were kept in 250 liters aquariums, control and experimental groups with the forces aeration and water purification. The hydrochemical regime in the aquariums was optimal in terms of the main parameters apart from the high water hardness and rather low temperature. Because of this, the weight gain of the fish was small. The fish was fed with the combined feed. The feed dose was calculated twice a week as 5% of the fish weight. For the experimental group, the feed was sprayed with a suspension containing subtilis S powder at the rate of 1 gram of probiotic per 1 kilogram of feed. After that, the feed was dried and fed to the fish. In addition, Chironomita larva was included in the diet of fish in both groups, depending on their consumption. The crayfish was kept in 2 aquariums, control and experimental groups, with forced aeration and water purification. The volume of water in the aquariums was 200 liters. In each aquarium, shelters made 2 ceramic tubes with a diameter of 40 millimeters were installed because they are cannibals. This showed in the slide hydrochemical parameters of aquarial water in experimental crayfish. The feed was medium-sized frozen bloodworm. For feeding the crayfish of the experimental group, the probiotic subtilis S was added to the bloodworms and the rate of 1.5 gram per 1 kilogram of feed. Crayfish were marked individually by applying nail varnish of different colors to the exoskeleton. The weighting was carried out on the electronic scale. Measurement of crayfish were carried out using a caliper. Blood was taken from the tail vein of the fish in vivo. Hemolymph of crayfish in vivo by ventral sinus punction. In this slide showed cells of circulating fluids of hydrobionts sending by popping gain. Carp, erythrocytes, leukocytes, and platelets of carp. This is hemocytes of crayfish, 4 tips hemocytes. Crayfish hemocytes in Горяевка chamber. Different counts. the average cytochemical coefficient was calculated by the formula cytochemical coefficient. This lysosomal cationic protein with reaction with bronchional blue different counts in different degree of activity. Cationic protein. During the experiment, the mildness of water in aquarium with the fish increased during the dry feed. As a result of this, as well as increased water hardness, white bloom appeared on small areas on the skin of 3 fish in the control group. Microscopic analysis of scarping from the affected skin areas of fish developed the presence of HEPA and spores of the infection agent Saprolegnia parasitica. In the experimental group, skin lesions were not detected, which indicated a higher resistance of fish in the experimental group to unfavorable environmental factors. After 2 months of feed in fish in the experimental group, achieved a significant increase of body weight by 48% in contrast to those in the control group. You might see that erythrocyte coined experimental group after 2 months increased hemoglobin increased in experimental group 2. Metamyelocytes in VBC differential is increased. So erythropoiesis in this group is activities. ACC of lysosomal cationic protein in neutrophils significantly increases in carp in the control group. On the contrary, the experimental group, there are no significant changes in ACC. It could be hypothesis that the activation of leukopoiesis in the experimental group was due to exhaustion of neutrophils and cationic protein in the lysosomes of the cells for immune defense. Quantity in crayfish after 1 month experiments, quantity of hemocytes increased in experimental group. Control groups. Что сказать аналогично? В общем, аналогично рыбам, analogy experimental of fish, ACC, cationic protein of crayfish also decreased. And it should be noted that crayfish were in unfavorable conditions for the first 2 weeks of the experiment, since during this period there was no water purification in the aquarium. In addition, the test individuals were carriers of the fungus, the Prolegnia parasitica, which causes burn spot disease. At the same time, the survival rate of crayfish in such accidentally extreme conditions in the experimental group was 2.7 times higher than in the control group. So it was found that the inclusion of the probiotics subtilis S in the crayfish food enhance the immune system survival in the presence of the Prolegnia parasitica in water, the incidence of rash spots disease in reduced. At the same time, the content of lysosome cationic protein in hemocytes decreased. Apparently, the hemocytes consume this toxic protein in the process of immune defense. Carp, which received a probiotic, also increases immunity to the prolegnosis, increased body weight gain, the amount of hemoglobin, and red blood cells. The fish leukogram showed an increase in metamyelocytes. Thank you for your attention.
Thank you very much, Professor Pronina, for very interesting communication. I wish you success. Let's moving to another participant. Я предлагаю выступить Андрею Дубровину. Так как у нас докладчик Юлия Маркова записана на видео, а ее показать позже. Андрей, вы готовы? На месте. Андрей Дубровин, это сотрудник фирмы Биотров, young scientist, expression of immunity genes of broiler chicken under Salmonella enteritidis infection. Thank you. Please.
Меня слышно? Здравствуйте.
Да.
Отлично.
Слышно хорошо.
Hello, dear colleagues. I'm glad to. I'm sorry. Just a moment. All right. Do you see my presentation? Hello?
Yes, okay. Okay, Андрей. Хорошо видно.
All right. Hello again, dear colleagues. I'm glad to present you the results of our research called the expression of immunity genes on broiler chicken under Salmonella enteritidis infection. Today, salmonellosis remains one of the most significant and dangerous bacterial diseases in the poultry and livestock farming. Salmonella remains dangerous not only to animals but also to humans. At the same time, Salmonella enteritidis remains the most prevalent, being one of the most common causative agents of salmonellosis in the European Union, Russia, and the United States. One of the challenges in the fight against Salmonella lies in the increasing antibiotic resistance, which is rising with the frequency of antibiotics use in agriculture around the world. New generations of antibiotics appear, but soon enough, Salmonella gains resistance to them too. At the same time, the productivity of new bird crosses is also steadily increasing, which affects the weakening of the protective functions of the poultry and facilitates the access of pathogens. These situation make us to search for new methods of protecting the poultry and strengthening its health. One of the solutions is the use of probiotics with bactericidal action against pathogens. They are being used widely in agriculture, but there is another interesting solution, the use of essential oils. They not only got a direct bactericidal effect against pathogens, but also may enhance the immune status of poultry, regulate inflammatory reactions, and stimulate their digestion. It is known that the body responds to the impacts at the molecular level, also through gene expression. It is the process by which information from a gene is used in the synthesis of a functional gene product that enables it to produce end products, protein or non-coding RNA. And it ultimately affects phenotype as the final effect. Gene expression is the most fundamental level at which the genotypes give rise to the phenotype. Such phenotypes are often expressed by the synthesis of proteins that control the organism structure and development, or act as enzymes catalyzing specific metabolic pathways. For example, in response to macrobiotic inoculation, the poultry body changes the expression of more than 250 genes. In chicken, Salmonella is initially recognized in the cecum by TLRs, followed by the subsequent induction of genes associated with the synthesis of hemokines, cytokines, and many effector genes that form the basis of the innate immune system. This leads to infiltration of heterophils, macrophages, and lymphocytes. Infection of chickens with Salmonella usually is characterized by the induction of an inflammatory response and an increase in the expression of many genes. But it was noted that the level of expression of other genes may be reduced. For example, infection of a 5-month-old chicken with Salmonella enteritis resulted in a decrease in the expression level of 32 different genes in the ceca and liver 10 days after infection. But these genes mainly belong to two functional categories, control of metabolic functions and the cell cycle. So the research field of salmonellosis in poultry is rather wide. And so the teams of Biotrof Ltd and our WIPS Institute have set an experiment on broiler chicken with the infection of Salmonella enteritis strain in the vivarium. Also, a feed additive based on essential oils effect on chicken was analyzed. The total of 120 chicks were raised in a vivarium from first day to the 42nd day, divided to the analogous groups, control group and experimental group. At the age of 19 days, half of each group chicks were infected intramuscularly into the pectoral muscle with the epizootic strain of Salmonella enteritis. and then further divided into control, infected control, experimental group, and infected experiment. The essential oils mix called the Interbio was added to daily feed from the first to the last day of the experimental group at a dose of 90 milligram on kilogram of compound feed. For laboratory analysis, samples of cecum tissue were taken a day and 3 weeks after infection. So this is a general outline of the experiment. In total, there were 4 groups of chicken, 30 chicks in each group. Laboratory research of the samples was carried out in Biotroph Ltd. In its molecular genetic laboratory. To analyze gene expression, eukaryotic RNA was isolated from the cecum tissue. A reverse transcription reaction was performed to obtain cDNA, and then quantitative polymerase chain reaction was performed. The calculation of the relative expression was performed using the double delta CT method by Levak and Schmidgen. In the early stage of infection, the use of essential oils helped to enhance the immune response in chicks in response to the infection. The expression of the genes of the cytokines interleukin 6 and interleukin 8 increased by 1.5 times 1 day after infection. In the experimental groups, the expression of interleukin genes increased relative to the control, both in infected groups by 4-6 times and intact experimental groups by 3-5 times. Expression of avian beta-defensin 10 after infection increased by about 2 times, and avian beta-defensin 11 decreased to 7/10 times compared to the control group. The addition of essential oils led to an increase in the expression level of the defensin genes, avian beta-defensin 9 and avian beta-defensin 11 by 1.5 times. The expression level of avian beta-defensin 10 increased 17-fold in the experimental group and 2-fold in the infected experimental group. The expression of the caspase-6 gene, it is a cysteine protease that plays a role in apoptosis regulation, increased 2.5 times in the experimental and infected experimental groups. The use of essential oils increased caspase-6 3 times in the infected control group compared with the control group, and 9.5 times in infected experimental group, 6.5 times in the intact experimental group. By the 3 weeks after infection, in all groups, the expression level of observed genes decreased compared to the control group, with the exception of avian beta-defensin 10 expression, which was still higher in infected control by 2 times, in infected experimental group by practically 4 times, and in experimental group by practically 3 times compared to the control group. I'm going to slightly expand the actual topic for more complete information, if you don't mind. Our team have also analyzed the microbiota of the ceca of chick using the TRFLP method. This method makes it possible to study entire communities of microorganisms by the bacterial 16S RNA region. Today, this may be not the most modern method of microflora research, but it is gaining popularity due to the speed and low cost of research in comparison with the NGS method or mini-ion research. These graphs demonstrate the composition of the microflora for the main phyla. 4 bars on the left indicates the state of microflora a day after infection, and 4 bars to the right shows the microflora 3 weeks after infection. So the bacterial community of the Broilus cecum a day after infection with Salmonella enteritis in almost all groups was represented by the Phylobacteroidetes, Firmicutes, Actinobacteria, Proteobacteria, and uncultured bacteria. A day after infection of broilers, in the cecum of infected control group, there was a sharp increase in the number of phylum Proteobacteria against a decrease in the content of uncultured bacteria, and also Phyla Firmicutes and Bacteroidetes compared to the non-infected groups. In the groups without infection and in the infected experimental group, the bacteria Firmicutes had a numerical advantage. 3 weeks after infection, the microflora balance in infected groups stabilized. Here you can see the picture of the 2 main taxa, Proteobacteria to the right and Firmicutes to the left. A day after infection, in the control group, there was a sharp drop in the number of genera Lactobacillus and Bacillus in comparison with the non-infected groups. At the same time, in the infected experimental group, no sharp differences from non-infected groups in the content of taxa of the phylum Firmicutes were found. A day after infection, a sharp increase in the number of bacteria of Enterobacteriaceae family in the infected control group was found, which indicates a competitive advantage in the intestine of Salmonella enteritis, which is the part of this taxon. At the same time, in the infected control group, there was a sharp drop in the number of representatives of the genera Lactobacillus and Bacillus in comparison with the other groups. So the obtained data indicates that the use of essential oils helped to enhance the immune response in different broiler chickens. But also our team carried out the same experiment on adult laying hens of Lomon White. I want to make a short brief about it because the results were a bit different. The age of the bird was more than 300 days. 3 weeks after a start of the experiment, the infection with the Salmonella enteritis was performed, after which the further division into control, infected control, experimental group and infected experimental group was carried out. Also, we still used the mix of essential oils to the experimental group. The sampling of the ceca was carried out 1 day and 1 week after infection. In the control infected group, 1 day after infection with Salmonella enteritis, a decrease in the level of expression of interleukin-6 gene was observed in comparison with the control intact group by more than 2 times. At the same time, in the experimental infected groups after infection with the Salmonella enteritis strain, the expression of the interleukin-6 gene significantly increased in comparison with the uninfected group and was about practically 2 times higher than the level of the intact control group. This trend continued and 6 and 7 days after infection. The fact that the level of expression of the interleukin-6 gene was the lowest in experimental intact group 1 day after infection seems logical and may be due to the absence of inflammatory processes. At the same time, an increase in the expression of this gene in infected experimental group indicates an activate reaction of the body in response to the introduction of the pathogen. At the same time, a decrease in the expression of this gene in infected control group in comparison with an intact analogue probably indicates a weakened immune system response. The expression of avian beta-defensin gene, it also called galenocin, 1 day after infection in all groups was lower than the control level, which may be more likely be due to an increase in the expression level in the control group. At the same time, the tendency of the variation of the expression of avian beta-defensin 10 after infection with the Salmonella enteritis strain between the infected control and 2 experimental groups correlates with the expression of interleukin-6 gene 1 day after infection. And 1 week after infection, an increase in the expression of avian beta-defensin 10 gene was noted in all groups relative to the control group. So it seems that the activation of this gene in adult hens occurred later than a day after infection with Salmonella, not likely in chicks broiler. The interleukin-8 gene is associated with the synthesis of hemokin that is involved in signaling between immune cells. 1 day after infection with Salmonella enteritis, in all experimental variants, the level of this gene expression was significantly higher than in the control group. The expression of interleukin-8 gene in the infected control group was 7 times higher than in the intact control. At the same time, the expression in the experimental group in comparison with the pure control was 14 times higher in the intact group and 15 times higher in the infected group. And a week after infection with the Salmonella, the slight decrease in level of expression of this gene between the experimental groups was observed. The higher level of expression of this gene in the experimental uninfected groups seems logical, since due to the function of this gene, it's increased because the increased expression of it may indicate a higher readiness of the organism to show an immune response in the event of pathogen entering. So the received data may indicate that the application of the essential oils to adult hens led to a more stable state of poultry's health with signs of higher readiness to an immune response. And 1 day after infection of the Salmonella enteritis was observed to more efficient immune response. Well, thank you for your attention.
Спасибо, дорогой Андрей. Thank you very much for very interesting report. I think maybe anybody have some questions, but this is a deficit of time because we are waiting, and I may answer all your questions about this report. Thank you, Андрей. Спасибо большое. А сейчас мне бы хотелось, I would like to announce Юлия Маркова, профессор Юлия Маркова, senior researcher, Federal Research Center of Nutritional and Biotechnology, Russia. Very interesting communication, development of a method for determination of probiotic microorganisms in multi-strain probiotics. My experience is this is a very interesting theme in practical sense.
Hello, my name is Yulia Markova. I work at the Federal Research Center of Nutrition, Biotechnology and Food Safety in Moscow, and I'm grateful to organizers for the opportunity to participate in the conference and to present our results. My presentation is related to the work on the method development for the determination of probiotic bacteria in multi-strain probiotic products. In Russia and Eurasian Economic Union, the definition of probiotic microorganisms as a food ingredient is given in the technical regulation of the Customs Union on food safety, and these terms include live, non-pathogenic and non-toxigenic bacteria, representatives of the protective groups of normal intestinal microbiota of a healthy human, and these ingredients are included in food products to improve the composition and biological activity of protective human gut microbiota. And in addition, only species of the lactobacillus, bifidobacterium, lactococcus, streptococcus, and propionibacterium genera are allowed for food purposes. There are some criteria for the effectiveness of probiotic products. First is identification to the genus, species, and strain level. It's important and should not be ignored because the positive beneficial effects of probiotics on health are strain specific. The second criteria is the amount of viable cells of probiotic bacteria in the product at the end of its shelf life, because the effect of those the effect is determined by the dose of probiotic bacteria. And the third criteria is the evidence for functional effects of probiotics in controlled experiments. Therefore, the identification of probiotic strains and their quantification in the products are very important aspects of quality control of such products. In Russia and in Eurasian Economic Union, there are approved state standards for the detection and quantification of probiotic bacteria. These methods are based on the traditional culture analysis with identification by biochemical tests. But in most cases, these standards only allow to determine the total number of bacteria and to identify bacteria only at the genus level. And these methods doesn't allow to detect, count, and identify individual species and strains of probiotic bacteria, and this is especially important for multi-strain compositions. At present, food products with probiotic bacteria hold a large market share and are in great demand, and at the same time, a large proportion of the currently produced probiotic products are multi-strain, and they contain several different lactobacillus or bifidobacterium species in one sample. Thus, the actual problem is to develop a method for identifying of probiotic bacteria in mixed cultures, allowing the identification and quantification of each of the added probiotic bacteria. This made it necessary to develop an accessible, reproducible, and highly specific method for the analysis of probiotic bacteria in various food products. The method choice was based on such criteria as specificity, the possibility of quantifying, and the accessibility. Molecular methods based on DNA sequencing are the most accurate and widely used method for microbial identification at the genus, species, and even strain level in the scientific research. However, such methods are labor-intensive, time-consuming, cost-consuming, and are not widely used in routine research. As a result, a PCR-based method was developed to quantify specific species of Lactobacillus and Bifidobacterium genera when their content is not less than 10,000 coliform units per gram. The steps of the method development are analysis of the scientific literature and selection of primer pairs. The next step is in silico verification of primer specificity and selection of species and genus-specific primer pairs. First, we analyzed the scientific literature and selected 204 pairs of primers specific to different gene regions that were genus and species specific to representatives of the Lactobacillus and Bifidobacterium genera. To obtain correct results and ensure a specificity of PCR, it's necessary to check the primers for suitability for the analysis in mixed culture. And we checked the specificity of primers in silico using the primer blood service by counting the number of homologs in two databases with a search restriction on the domain bacteria. And based on the results of the check, we excluded primers that didn't meet the necessary requirement for adequate PCR analysis. And as a result, 26 primer pairs specific to most widely used species in probiotic products were selected, including primers specific to 16 Lactobacillus species and to 7 Bifidobacterium species. And the optimum conditions for PCR assay with typical text traits of Lactobacillus and Bifidobacterium bacteria were selected, and we established the detection limits for each pair of primers, and the whole algorithm for the detection of probiotic bacteria includes several steps. The first step is detection and quantification of viable bacteria by inoculating of tenfold dilutions of the sample into a culture media with following incubation. The second step is DNA extraction of target bacteria without isolation of few cultures directly from the culture media. The next step is direct identification of target bacterial species in the DNA extracts by PCR using genus-specific and species-specific primers. And the next step is quantification of target probiotic bacteria. And with the use of the developed method, we tested 67 samples of probiotic dairy supplements, including 32 multi-strain samples. These multi-strain samples contained up to 14 types of probiotic bacteria in one sample. And it was found that 62% of the multi-strain samples didn't meet the specification regarding the total number of probiotic bacteria. 50% didn't meet the specification regarding the number or the amount of individual probiotic strains, and 53% regarding the species composition. And the developed method formed the basis for the draft guidelines, methodological guidelines for the identification of probiotic and technological bacteria in food and dietary supplements. This method makes it possible to detect and identify not only live probiotic bacteria, but also a non-viable cells of probiotic strains. And in addition, I find it necessary to note that in 2020, the genus Lactobacillus have been reclassified reclassified into 25 genera, the amended genus Lactobacillus, and 24 novel genera. And in accordance with this reclassification, 2 services have been created, and these services can help to verify the genetic names of bacteria, of lactic acid bacteria, and to translate former names into a new one. And this information should be taken into account in following research. Thank you for your attention.
Thank you. Thank you very much, dear Professor Markova. Dear colleagues, our program is over, and till в общем, я решаю объявить перерыв. Till 14:30. Yes, let's have a little rest. Так, я выключаюсь, да?
Просто они сейчас разойдутся, и что мы будем делать? Может, имеется в виду, что здесь...
I shall repeat, dear colleagues, we started after the rest in 14:30.
1, 2, 3. Один, два, три.
Hello, can you hear me? Yes. Good afternoon, everyone, and the very good evening for those of you who are across the planet. I'm so happy to see Slavy Todorov, Minze, Dzongdib, and others. Let me just put on my, yes, Santosh. So it's an old guard, as we say. Before we start the official ceremony of closing this absolutely fantastic conference, I would like to spend some time announcing the Young Scientist Awards. This conference was really interesting in many ways, and one of them was that in addition to the old-timers as myself and Slavy, so there are two old-timers, there were many young scientists, young investigators, PhD students, those who are just in the beginning of their professional career. And it was particularly interesting, fascinating to see how this new next generation of researchers are involved with their work and how enthusiastic they are. So keeping up with the tradition of the conference, which was started by Dr. Leong some years ago, we also have the Young Investigators Award this year. And Dr. Deep, I must say I was jealous seeing how you were packing some bags for the participants. So I insisted on myself also being a part of the same thing with this conference. So I know how it feels, yes. So without any further ado, I will start announcing those who are with us and those who are not with us. One award was already given to Dr. Tsavieva, who left to catch the plane to St. Petersburg. So I will go with those whom we have here, and I will also announce those who are not with us but across the globe. Of course, they will receive their awards. They will come to them by mail. I would like to start with the first person, Alexei Vatlin. This is yours.
Thank you very much.
And thank you for your outstanding presentation.
Thank you.
Thank you. The next awardee, Alena Abayimova. And thank you for a great presentation. It's a particular pleasure for me. The speech was very interesting, but the place where this young researcher came from is the place where I used to work right after I finished my PhD last century. The third person who was also noticed by our committee is Dr. Maria Mazenka. And everybody noticed particularly the enthusiasm and the emotional drive when Dr. Mazenka was presenting her speech today. I would like to also announce a few more awardees who are not with us, but they will, as I said, they will receive their awards in no particular order. Jorge Enrique Vasquez Buccelli, from South Korea, Joanna Ivy Yorita Fogabanan, also from South Korea, and Rivaz Lapachi from Georgia. And we will make sure that the awards will reach these investigators, and we're particularly touched by the fact that many young scientists, literally from across the globe, were so enthusiastic and shared with us their research. Thank you very much. I would like to ask the rector of the Don State Technical University, Professor Meschi, to say a few words of concluding remarks for our conference.
Спасибо большое. Я эти несколько дней наблюдаю за нашей конференцией, принял участие в открытии и послушал даже несколько докладов.
I'm following this conference from the very beginning. I was making the opening remark, and I also had a chance to listen to a few of the presentations.
У нас приняли выдающиеся ученые нашей страны, которые дали возможность и самим выступить на этой конференции, сделать свой доклад. И я думаю, что хочу отметить особо, сегодня присутствует у нас Юрий Анатольевич Иванов, академик Академии наук, Российской академии наук.
I would like to bring your attention to the fact that the very outstanding Russian scientists were presenting their talks at this conference, but also today we have a pleasure of seeing with us Professor Ivanov, who is an academician of the Russian Academy of Sciences.
И как раз ученые такого уровня, которые приняли участие сегодня в нашей конференции, придает особый статус, особый уровень и необходимость такой конференции.
And the quality of the scientists, the caliber of the scientists who participated in this conference actually is a true flavor of this event, and they're making it particularly noticeable.
Сегодня была рабочая встреча с учеными из Санкт-Петербурга, которые приехали к нам. Мы обсуждали вопросы о сотрудничестве, о тех результатах, которые уже получены на данной конференции, где мы будем работать и сотрудничать.
Today we've had a special meeting with two scientists. Some of them were from St. Petersburg, some from Moscow, and we were discussing the opportunities for future collaboration based on what was reported during this event.
Интерес данной конференции, еще раз отмечаю, что это безопасность страны, безопасность людей, безопасность питания и всего то, что сегодня нас окружает, в принципе, вы сегодня обсуждали.
I cannot stress enough saying that one of the major topics for this conference was safety and security for people, safety and security for the countries, safety and security for the food and for our environment.
Еще раз огромное спасибо профессору Чекиндас. Юрий Анатольевич, вам огромное спасибо за то, что вы нашли возможность приехать на данную конференцию и сегодня своими глазами увидели именно заинтересованность участников данной конференции. Я хотел бы еще раз подчеркнуть, дорогие друзья, дорогие наши коллеги, которые участвуют из разных стран, приглашаем вас в сотрудничество, работать вместе, и Донской государственный технический университет открывает двери для вас, для всех.
I want to repeat that we are particularly grateful to academician Ivanov for finding some time in his very busy schedule to be with us, and so he can see himself how enthusiastic, how involved all of us into these events, those who are here physically and those who are distant from different countries. I want to stress again, to emphasize that we would like to welcome all of you, and the doors of our university are open, and we hope to see you when it will become possible here in Rostov.
Как всегда, когда начало конференции, все радуются. Когда закрывается конференция, все грустно смотрят друг друга, потому что мы расстаемся. Но надеюсь, что те вопросы, которые мы наметили с вами, это будет как раз продолжением той работы, которую мы с вами в этот раз обсуждали. Придется работать молодым ученым, которые здесь присутствуют, продолжать все эти данные вопросы, которые мы обсуждали здесь. Декану факультета Рудому тоже даю поручение. И, конечно, лаборатория, которая у нас создана сегодня под руководством профессора Чикиндас, я думаю, что продолжит работу, и мы будем продолжать данные вопросы, которые мы обсуждали. Удачи нам всем еще раз. Мы не завершаем работу, мы продолжаем работу.
It's always the same case when any event starts. Everybody is happy, smiling, and whenever any event, especially the good quality event, is ending, everybody feels sad because it means that we have to say farewell to each other. But this is not the end. This is just the beginning. And we hope that the foundation which was laid during this conference will continue to grow. I'm not saying goodbye, I'm saying that we are just starting to work, and I look forward for new performances, new achievements in the future. I'm not saying goodbye, I'm saying we'll see each other soon again.
Спасибо. Добрый час.
Thank you, farewell and Godspeed. I would like to give the floor to our honorable guest, academician of the Russian Academy of Sciences, Professor Ivanov.
Thank you. First of all, I would like to say a great thank you all participants this innovation conference. I would like to say one more, innovation conference, for interesting and useful reports and presentation. Also, let me inform you about our priorities Russian scientists. I mean agricultural sector of Russian Academy in this topic. First of all, it's biological land cultivation, because it's first stage in produce organic food. Also, our scientists still pay great attention right now and the future, because for its main goal, provision our population, quality products in sufficient quantity. I hope this meeting conference will be continue in the future. Thank you.
And as the final glory point of our meeting, with great pleasure and honor, I am giving the floor to the founder and president of our conferences, and I say in plural because this is a chain of conferences which will continue with no doubts. Professor Minzeng Leong, please.
A very good afternoon, everyone. It is my pleasure to be among the best. Very good afternoon to Professor Meshki, Rector of Don State Technical University. And I can see here today we have Professor Ivanov. Thank you very much for coming, all the way from the Russian Academy of Sciences. My greatest gratitude actually this time to Professor Michael Chikendes. So as I've mentioned in my opening, we started in 2014. We have this conference series once every 2 years. So 2014 was in my university, University Science Malaysia. 2016 it was in Phuket, Thailand. 2016, after that, 2018, we had it back in Malaysia in a city called Kuching. And 2020, we wanted to have it in Taipei, Taiwan, but because of the pandemic, it was completely canceled. And at that time, it broke my heart. But Michael, as the savior, came to light and he told us, don't worry, we will bring it to Russia. And I have never been to Russia myself. So I was so excited. And with the pandemic again, I cannot come. So I do wish to come one day. And I thank you for your welcome to the university. Now, as you can see, we have a very good chain of speakers. We always have. I personally hand select all my speakers. And it turned out that we became family. And now with Russia, and I see how the Russian team has brought this event to a record-breaking scale. I repeat, it is 387 participants from 87 countries. We have never hit 300, and now we have exceeded that. And when I painted in my opening ceremony the number of countries that participated, we covered 3/4 of the globe. So the Russian team, I am more than impressed. So Dr. Dmitry Rudoy, you said that you hope you did not disappoint me. No, far from that. I was walking on clouds when I saw the numbers. I was so happy. And when I saw the Russian scientists presenting their work, I want to collaborate. I have seen this quality, top-notch quality, and indeed I want to collaborate. So I hope, Michael, you can link us up. And now with this scale, we will continue this ICOM series and we will add the Russian into our family. So welcome to our family. And for 2023, 2 years from now, we have yet to decide the venue, but I hope at least some of you will be able to join us again. And lastly, to all the young scientists, I hope you have benefited from this event. Like I said, ICOM is a conference, a very special conference, because most of the funding that we always get, we always give it back to the young scientists. So this time I see the young scientists are also performing very well, presented their work again, top-notch quality. So I'm very proud, and it's my pleasure and honor to have met all of you, and to all the old members like Zong, like Slavi, Santosh also recently met. Welcome to the family, and let this tradition continue. So I won't hold the floor longer. Ladies and gentlemen, thank you very much, and see you again.
Thank you, Minze. Thank you all of you who are still with us, because we're, as I mentioned, all across the globe, different time zones, different, you know, really different from like midday to the late night right now. And again, you know, I'm getting emotional. We still have to say goodbye, but I'm saying that I'm looking forward to meeting again. 2023 is just around the corner. So we'll definitely see each other, and I hope that the young generation who presented beautifully today, some of them will be able to come and to join us at the 2023 event. Thank you, everyone, and we continue to work, and we continue to be collaborators and good friends. Thank you all.
Bye-bye. Bye-bye.
Bye to everyone.